Bonjour. Un, deux, un, deux. Bonjour. Un, deux.
Ouais, donc je peux enlever mon masque sur le... Je peux enlever mon masque.
Ah bah bien sûr.
Oui, oui, on va commencer. Il va apporter.
Hello everyone. Allow me to introduce our first speaker for today, Judith Mene-Hatab. She's a senior researcher with the compartmentalization and dynamics of nuclear functions team at Institut Curie Paris. She works on high-resolution microscopy techniques to explore DNA dynamics, and today she will explain how to image nuclear organization at high resolution. The floor is yours, Judith.
Okay. Thank you very much for the introduction. I hope you can hear me well. Okay. And thank you for the organizer for inviting me. It's really a pleasure to be here. It's my first time at MIFOBIO. So today I'm going to talk to you about imaging nuclear organization and DNA repair at high resolution, mainly by palm and single particle tracking. So we already know that the nucleus is organized in membraneless subcompartments where specific functions are achieved. So here is an example of a yeast nucleus where we can see already different subcompartments. Here, centromere clusters, silencing foci, repair foci, the nucleolus. And in contrast to compartments found in the cytoplasms, in the nucleus, subcompartments have no membrane. Inside this subcompartment, we found a very high concentration of specific proteins, for example here, silencing proteins or repair proteins. And we know that specific proteins can penetrate inside this subcompartment while others are excluded. And it's not only due to their size, it can be also small proteins that are excluded sometimes. So we try to understand how these subcompartments are formed and maintained. And of course, these subcompartments, they are not specific to budding yeast. They also exist. Here. They also exist in other organisms, even in human cells, as you can see here. So we try to understand what is the physics of these subcompartments, and is there a universal mechanisms to form these condensates. And to understand how these nuclear subcompartments are formed, we use single molecule macroscopy. And if you want more details, the first part of the talk that I'm going to present has been recently published. And at the end, I have a second part which is really small with unpublished results. So more specifically, we studied two types of subcompartments, repair and silencing foci. So for repair subcompartments, when there is a break, a DNA damage, so a break in the DNA, there is a high concentration of repair factor that form a very high high condensate, highly concentrated condensate, which we call focus. And the formation of this repair foci is extremely important for genome integrity. Similarly, we found also silencing foci here, where specific factors for silencing are concentrated, for example, at heterochromatin or close to the telomeres. And these silencing foci are extremely important to repress gene expression. And in the lab, we have tools to induce the formation of these foci at will, and we can also study their dynamics, their rearrangements, and we know that in certain conditions they can fuse. So I'm going to discuss some models of subcompartments that have been described in the literature recently. So in a very simple model, these subcompartments are formed just due to the binding of specific proteins to chromatin, as illustrated here. So in this model, which we call the binding model, we don't have a very high accumulation of protein at this specific DNA site, and there is no phase separation. In contrast, in the bridging model, there are protein-protein interaction, and protein can form bridges which will put very distant loci together and form loops. So in this model, we can create what we call the polymer-polymer phase separation, or PPS. Okay. And in the last model, that we call the droplet model, proteins self-organize to form this liquid environment, liquid droplet, where specific proteins can enter and others cannot enter, so they are excluded. So in the literature, this model has been described extensively during the last 5-10 years. So for example, there are these papers where they propose that Reaper foci in the nucleus, there are liquid compartments formed around a break around in the DNA. So they have mainly results using in vitro experiments or some in vivo microscopy, but not at high resolution. They also propose that heterochromatin is a liquid reservoir to silence chromatin, and there are many these two papers, which are very controversial, I have to say, proposing that heterochromatin is also a liquid-liquid phase separation. But in fact, the criteria to define these nuclear cell compartments are a little bit weak, I have to say. So basically, the liquid criteria are the shape of this condensate, their ability to rearrange inside, so the diffusion we can measure inside the foci, their ability to fuse, or their ability to be dissolved upon specific drugs. But all these conclusions are based on macroscopic observations, and there is a lack of in vivo evidence at the single molecule level. So today I'm going to present you some results on Reaper foci to show you how we use single molecule tracking, so SPT, to really prove that Reaper foci have liquid properties. So this is a simplified view of the biological process we looked at. It's DNA repair. So when there is a double-strand break in the genome, it can be repaired by different pathways. The pathway that we are going to talk today is homologous recombination. So it's using an intact homologous DNA, here in red, to restore the lost genetic information. And for that, specific repair proteins are recruited at the damaged sites. So in particular, the protein I'm going to describe today is RAD52 and RPA. So RPA cover the single-strand tail formed after DNA breaks, and RAD52 is recruited here, then to remove RPA and to recruit another protein, RAD51. And just for you, the human analog of RAD52 is BRCA2, which is involved in different cancers. So this is a book picture, but in vivo, without super resolution, it looks like this in yeast. You have RAD52 here tagged with GFP. And you can see that when there is a break, you have an accumulation of RAD52 proteins forming a very bright spot in the nucleus, which is here. So this concentrates a lot of proteins. But in fact, the size of this foci is extremely small. Because if we look at the yeast, the full yeast is about 10 micrometer. The nucleus is about 1 micrometer in radius, and the focus is much smaller. So typically it's under the limit of the light diffraction. So this is why we came to super resolution. And we used two approaches in collaboration with the lab of Maxime Dahan and Bassam Haj. So we studied the dynamic of this protein using single particle tracking in living cells to follow individual RAD52 proteins. And we also studied the structure and the size and the properties of these foci in fixed cells using PALM approaches. So to give you a little bit of technical details, we used different approaches. The first approach to do SPT was to use photon convertible fluorophore, for example, MAPL and MEOS, and we tested different constructs. And this was working relatively okay. We had a high density, but we had also, it was a little bit hard to do the SPT because when we have such a high density of fluorophore, we had a little bit of mislinking, and also the signal-to-noise ratio was relatively low with M-Maple and MEOS in yeast, at least. So we had lots of results with that, but we gave up. At the end, we used almost all the time another approach, which was to inject dye in our nuclei, or to incorporate these dyes into nuclei, and we used the JF dyes from Luc Levis. In particular, we had photoactivable JF dyes and fluorogenic dyes. So in most of the results that I will present here, it was done using fluorogenic dyes, which emit lights only once they are attached to a halo site. So using this technique, it was really nice to do the SPT, because we had a low density of signal and low density of detections and trajectories, and we had also longer traces, which was better to do further analysis. So we have to go to the microscope now. So these are yeast, and these are individual Rad52 molecules. They are actually they are all fused to a halo, and some of the molecule will bound to a halotag JF dye. And here is this is exactly what you see: individual molecules moving in the nucleus, and this is in the absence of DNA damage. So they are all dancing in the nucleus, waiting for a break to happen. And we can recover their trajectories like this. And so we detect, then we track, and then we try to extract some information such as diffusion coefficient, type of motions, residence times, etc. And then we try to understand the physical nature of the repair foci. The nice thing about this technique with incorporating JF inside the nuclei of yeast is that we can really tune the amount of molecules that we can see during an experiment just by changing the concentration of dye. So here I have a movie where it's a little bit dense. I have 10 nanomolars of dyes, but typically you can also use a lower concentration of dyes, which gives you something a little bit more boring when you look at it, but for the analysis it's actually better to be less concentrated. So I don't know if the second movie was running well, but actually the second movie is nicer than this one for the analysis. And here is what you obtain. So this will be the construct, a time projection of all the Rad52 molecules dancing in the nucleus in the absence of break. We do the tracking in 2D, like this. And when we have all the detections, we connect the traces, and we have this kind of graph at the end for a single nucleus. From this data, we can extract first the mean length of all the trajectories and the histogram of the length. So typically, to give you an idea, the mean length is 7.5 frames, so it's not so bad for SPT. And we can see that sometimes we have very long trajectory. Most of them are 2-3 points, of course. And then we extract the histogram of displacement for our protein, Rad52, in the absence of break. And we can fit this experimental histogram here in green using two models. One which gives us a one population model, which will allow us to extract the diffusion coefficient, the effective diffusion coefficient in the nucleus, and another model in which we assume that there are two populations that diffuse with D1, D slow, and D2, D fast. And what we saw immediately is that in our experiments, the data are well fitted with the two population model, as we can see here. And here is a detail where we show the fast and the fast in yellow versus the slow population here in purple. So that's the first very basic thing that we can learn. Okay, we have repair proteins, and they have two modes of diffusion, a fast and a slow. But the slow is it's not super slow. It's not like chromatin, it's relatively high, actually. And if we take this diffusion coefficient that we found, the fastest one, it means that in a simple Brownian model, to cover 1 micrometer, a RAD52 molecule would need 200 milliseconds. So this is approximately the time that a molecule would need to have to go through half of the nucleus. Okay, because this is the radius of the nucleus. And just to compare this number, the diffusion for RAD52, the fastest one, with a free molecule, the free molecule is a little bit faster, but not so much. And it also allows us to estimate the viscosity of the nucleus, which is 0.1 Pascal second. So it's a little bit between water and honey. So what can we say about these two populations when we do SPT? The first thing was to look at the literature, of course, and what we saw is that RAD52 exists in vitro in 2 forms, monomers and 11-mers. Okay, so from these structures we can estimate the theoretical diffusion coefficient of RAD52 from the Stokes-Einstein formula, and compare with what we found in vivo to see if it's the diffusion coefficient we have correspond with the proposed form of RAD52 in vitro. And actually we found that the estimated coefficient for a monomer and multimers in the theoretical one correspond to what we found with SPT. So it was really a good way to evaluate how RAD52 is, which form is existing in vivo. But now the interesting thing, of course, is to induce breaks, and this is what we want to do. So we induce a break. We use a tool that we have in the lab, IC1, a restriction enzyme that can induce a single clean double-strand break in the genome, and we know exactly where it is. And then the idea is to induce a break and to look at RAD52 mobility, so how these repair proteins are going to find the break, to accumulate at the close to the damaged site, and to form this huge membraneless subcompartment that we discussed at the beginning. So this is what we have with classical microscopy, and this is what we get with SPT. Okay, so we have RAD52 molecules accumulating at the site of damage, DNA damage, and we immediately see that they move slower than outside of the break. And to be really convinced, this is what we see. So these are single nuclei without break. These are single nucleus with a break. So this is the break. And if we look at the displacement histogram of RAD52 inside the focus versus outside of the focus, we see immediately that it moves really differently. Outside of the focus, molecules, they move exactly as they were moving before breaks. And in the focus, they move slower. To go further, we also wanted to understand how do they move, what kind of motion they have. So we used mean square displacement analysis. So it tells us the mean distance covered as a function of the time interval, and we can use different models to then fit the experimental MSD. And what we found is that inside a foci, if we just look at trajectory in these subcompartments, we have a MSD exhibiting a plateau, a clear plateau. So it tells us that molecules, they are confined inside these subcompartments. And we have the confinement radius, which is 120 nanometers, relatively small, and the diffusion coefficient. Interestingly, we also perform palm experiments to measure the size of this foci. So it's a totally independent experiment, different strain. It's actually a strain with M-maple, RAD52, so it's not the JF system and the halo. And we measure the size using palm, and we found that the confinement radius found by SPT really matches with the size of the foci found by palm. So in other terms, when we have a repair focus which is formed, molecules inside the focus are exploring the whole subcompartments. They are not really stuck on the single strand tail. Okay. And So this is our view. So this is the nucleus. This is a repair focus, so the subcompartment where we do repair. And inside you see individual repair molecules. And we compare the mobility of the individual molecules with the mobility of the whole focus. Because, of course, these compartments, they can move also a little bit by themselves. But we saw that inside, individual molecules inside the foci, they are much more mobile, here in red, than the whole focus itself. So we really have to see this as a small compartment moving relatively slowly, and inside its molecules are really dancing fast. And this is what you can see using the MSD, because with the MSD of individual molecules inside foci compared with the MSD of the whole focus, we can immediately see a very striking difference. So going back to models, well, in a simple bridging model, we would expect inside the subcompartments to have a relatively low diffusion coefficient, similar to the substrate, so chromatin, and to have also eventually a second population of molecule moving when they unbind a binding site, and then they move like free molecules outside. So this would be a relatively normal explanation for this model. And we also expect to have a kind of motion for individual molecules inside foci like chromatin. So at that time scale, it would be anomalous motion. But this is not what we saw. What we saw is that inside the focus, we had one population, one diffusive population, and it was moving relatively fast compared to chromatin. And molecules were really exploring the whole focus with the confinement radius matching with the size of foci. So this is the first conclusion that we get from SPT experiment. And it was kind of unexpected when we tried this at the beginning. And to make sure that we were not wrong, we also looked at another repair protein which is really expected to stay stuck at the DNA damage site. So for that, we looked at RPA, which is a protein always bound to the single-strand tail. It's called a single-strand binding protein, actually. So it forms also foci, similar to RAD52, so similar subcompartments. We looked at it with SPT, and what we found is that this new protein is moving less fast than RAD52 inside the foci. It's moving like the whole focus and like chromatin. And when we look at the MSD, it shows exactly the same MSD than chromatin. So this is a kind of control for us, because we use exactly the same technique and we see very different properties as we expected. So a very interesting approach is also to look at how molecules behave at the boundary of repair foci, or any subcompartment would be quite interesting. Because actually these subcompartments, they are not closed. They don't have a membrane and they are not closed, so molecules, they can escape sometime, relatively often in the case of RAD52. And we looked at the way they move. So we concentrated on the traces that cross the boundary at least once. And using SPT approach, we really saw protein moving from inside to outside, or the opposite. And this is what we see. So these are displacement of RAD52 in micrometers inside the foci when they cross, and outside. And this is molecules that go from outside to inside. Okay, so after we accumulated a lot of data, we just concentrate on the red protein that cross the membrane and plotted this distribution of this displacement of RAD52, and we found that, interestingly, when they cross the boundary, they start to move super fast, and when they enter inside foci, they slow down relatively fast. So we have a sharp decrease of mobility as soon as it enters inside the subcompartments. So these are two interesting results obtained by SPT. And now I'm going to discuss another criteria that is often used to define liquid or not liquid subcompartments. It's their ability to fuse together. So to be clear, when there are multiple breaks, you would expect, for example, to have 3 breaks and then to have 3 repair foci formed. But in fact, when we looked at RAD52, after a few minutes, we can see that we have only a single repair compartment which is formed. So it was proposed that repair foci, they transition to 1 or 2 foci, even when there are multiple breaks. The same observation has been done also in human cells, although it's only foci close enough which are able to fuse together. So we have done this in yeast using our system with SPT. So the idea was to induce 2 double-strand break with 2 RIC 1 cut site this time, and to look at RAD52 using SPT again. And indeed, here are individual nuclei. So it's just a time projection of all the detections. And we can see here in light blue the foci that are formed. We have 1 focus per nucleus, as we can see here. Okay. And in that case, we measure the size of the foci. And foci formed after 2 double-strand breaks are larger than 1 double-strand break. And then we ask, what is the internal dynamic of this foci? And this is something we can really see by SPT. So if we have an individual break, we can look at how individual RAD52 molecules move inside. But then if we have multiple breaks, do they move independently, the different subfoci, or do they really fuse together as we would expect in a droplet model? So to distinguish between these 2 scenarios, it's possible by Palm because we can plot the MSD of RAD52 trajectory inside this foci and see if the confinement radius increases between 1 and several breaks. In this model, it will be the same, and in that model, it will increase. And actually, what we saw is that when there is also only 1 break, the MSD is plateauing at a lower level than when there are 2 breaks. Okay, so this is again a view of the whole focus and an individual trajectory of repair proteins inside. And when there are 2 breaks, the focus is larger, and molecules they are able to explore these larger subcompartments. Okay. So this is another evidence that repair proteins they are not always bound to the DNA damaged site, but there are lots of proteins actually in these subcompartments are really free to move relatively fast. They are not bound, and they can explore the whole repair focus. So this is something that really goes into that kind of confirm the droplet idea. And more recently, we also have a collaboration with physicists at the ENS, and they perform simulations that about the bridging model and the droplet model, and they plotted this curve showing that the density inside and outside foci versus the diffusion coefficient inside and outside of the foci, and what they found for different value goes on this nice slope, and the experimental observation that we have is really far from what we expect here in a simple bridging model. So it's again a confirmation that for us that we can reject the bridging model in the case of repair foci. So just to finish this part, we set up a system in budding yeast to really look at individual repair proteins inside living cells. We are also doing this now on silencing foci, but it's in progress. We saw, surprisingly, that RAD52 molecules, they are slower inside the foci, but they are relatively, they are more dynamic than what we thought. They have confined motion and a high rate of exchange with the rest of the nucleus. And finally, we found that multiple foci, they really fuse together. They are not just closer, and it's not just a colocalization and they are a little bit close. They really fuse together, and molecules, they can be shared between the different subfoci and mixed together. Okay, so that's all for this part. And just to finish, I'm going to show you also another project which is also about DNA damage and SPT. But here the idea is not to look directly at DNA repair proteins, but to look at chromatin remodeling upon DNA damage. So this is unpublished and it's very recent. It's a work performed by a very talented postdoc, Fabiola Garcia. And the idea here is to look at the mobility of H2B histones in mammalian cells before DNA damage and then after DNA damage. So this is what we have. So these are individual histones. And these are the trajectories that we can find here from this movie. And this is all the trajectories, time projection of all the trajectories. We can already see that we have some long traces and some short traces. And just to illustrate something that happens sometime and which is not so good, in SPT, the crowding is really the enemy because we don't want to have too many detections. So, because it's also a course today, I wanted to show you something which is not that we shouldn't show, so something which is not working super well. But here, it's the kind of acquisition that you should not keep if one day you do SPT, because it's a little bit too crowded. Ah, the movie doesn't start. Okay, so in that case, what you get at the end is something like this, and because we are too crowded and we have too much background, it's not so good. Sometimes we have mislinking, and it's just to explain really with images that crowding is the enemy of SPT. It was easier to explain here on mammalian cells than on yeast, because it's super small, the yeast. So, what do we do with this now? We have developed, in collaboration with Sébastien Huet, who's going to talk just after me, and also in collaboration with a company called Erol Laser Company, we have implemented on the SPT setup another laser to do UV macro laser irradiation with this wavelength, which does not interfere too much with the photoactivation. So the idea is that we can do SPT, and at the same time, we can cut DNA along a line. So it won't be a single double-strand break, as I explained before with IC1, but it will be a line full of many DNA damages. And the idea was to look at the recruitment of a repair factor, so here it's PARP1 in mammalian cells, and then to compare the mobility in that specific slide of the histones before damage versus after damage. And this is what we get. The blue is before and the red is after. So using this setup, which is really something new that we have developed by ourselves with the help of Erol Company, we can really access the dynamic of histones right after the DNA damage in real time. Okay. And the idea to continue this project is to quantify the changes in histones mobility just after DNA damage, and then also to quantify how the PARP1, for example, repair factor are recruited, but right after we induce the damage. So really to see how they sense the DNA damage and how they go there with the SPT in living cells. So, saying that, I would like to thank all the people who contributed to this work. So, of course, the PI of the team, Angela, Marie Vilmer, who is an engineer and performed many strands for the project, Matthias, the theoretician who worked with me to analyze the SPT trajectories, Bassam, Chloe, who started the project really at the beginning, and the physicists, two physicists who collaborated with us on the theoretical aspect of the droplet models. So thank you very much, and I'm happy to answer your questions.
Thank you again, Judith. We pass to the question.
So, Judith, can you hear me?
Yes.
So, I was very intrigued by this separation that you found on the foci, and specifically about the boundary. Do you have more idea about what this boundary could be due to? For example, is it due to the fact that the chromatin has been reorganized, and then you have an increased chromatin over there, which create apparent boundary, so it's very hard for a molecule, for example, to get outside or inside? Or is there something else? And so the first question. And the second question is, looking at your acquisition delta T, from what you wrote, it's about 20 milliseconds, right?
Yes.
And so in 20 milliseconds, with 1 micrometer square per second outside, I don't know, you have like a mean square displacement, which is square root of 2d delta T, which is about hundreds of nanometers, which probably it's very large steps that you have. And so how can you study then entrance into a region which has more or less the same size? So that's my two questions.
Well, when they are inside the foci, they slow down.
No, but
No.
from outside to inside, because then it's very large jumps. So I don't know how many, for example, how many steps you have inside. Can you get, like you show a graph on this? So you have, just for example, this question could be, have you tried at 1 millisecond, and does your result change with 1 millisecond acquisition delta T?
No, we haven't tried. But what you mean is that if you enter inside a focus with this speed, you should just go on the other side.
That's
Actually,
my concern.
You could just traverse it and without feeling it.
I don't know. I'm asking because it looks to me very, very big. So in order to get more security about your data, you know, one way is to just decrease your time step delta T. So that's why you know.
Yeah, that would be interesting. But I guess as soon as they enter inside the foci, they slow down immediately. This is why they can get stuck. But it's possible that a percentage of molecules they are close to the boundary, but they just traverse the foci and we don't even see them. But that I can't see it. I mean, I can't measure it. I can't measure. Well.
So this is related to the first question. So what do you think about what is the boundary?
That I don't know. I guess there is a change in viscosity between the environment inside and outside, but I can't tell you right away. Oh, there is a change in the chromatin density in the focus, because I don't have a proof of that, an experimental proof. But that would be an explanation. We don't know exactly. For example, here, this is what we draw a little bit, because here we have a lot of chromatin, where here it's much less packed. That would be, but I don't have an experimental proof of that. This is why the second project where we looked at the histone and will be interesting, because we can really see if we have something more packed. Actually, it has to be measured also by storm, this kind of thing, just to have access to the structure. The structure of chromatin in versus out of the foci. Are you okay?
Yeah. No. Yeah. So, but you really think that there could be a change in the viscosity? Because I'm not sure we go from water to honey. I mean, the viscosity is probably the same. The crowding and the reorganization of crowding is probably what could mediate this. Effective boundary.
So a possible experiment to answer to your question would be to measure, for example, the density of histones in the nucleus and induce a break and see if I have a kind of a super high density of histones colocalizing with the Reaper focus. I'm not sure we have that, because I haven't done the experiment and I don't know. The thing that where we have the break, and when we remove the histones, so it's less packed. So I don't know exactly. Maybe it's not histones, maybe it's not chromatin, it's also all the other repair proteins which make this environment denser. Okay, let's discuss this
Sure.
after
Thank
also.
you.
Yeah, very very interesting talk. I have a question about it's maybe following the previous one about this phase separation, liquid phase separation. What would be from microscopy observation you would put as a criteria of phase separation? Because from point of view of a chemist, you can have a phase separation between organic phase and the water phase, and because they're not miscible. Here, what is the criteria why you have a phase and why you can call it phase? And can you define it with microscopy measurements and identify it as a phase or just as an aggregate or just as a complex?
It's a very good question. Actually, the parameters to really define the liquid phase separation, they are not totally clear. And a lot of parameters that are used, like fusion, they are used in many papers. They should not be used because sometimes you can have a polymer phase separation aggregate that fuse together and they are not liquid at all. They are just solid that go together.
So it's like polymer nanoparticles.
Yes, exactly. Okay. So for us, what we have tried to do is to go a little bit further, thanks to the SPT, and to say that, for example, if you use the fusion criteria as a liquid thing, you have to really have real fusion with molecule moving everywhere in a larger subcompartments, and not just something getting larger and closer. So this, for example, would be an important criteria. Okay. The fact that the molecule increase, the confinement radius increase. Okay, and also measuring the diffusion coefficient inside the foci and seeing that it's quite different from chromatin. It's also a criteria because it means that you don't, at a certain point, you don't really need the substrate to have the compartment because molecules they are here and they self-organize to form this compartment. They don't really need to be attached to chromatin. So it's another criteria.
And these structures never contain lipids, right?
Never contains what?
Lipids.
Not in yeast.
Because with lipids it would be easy to define a phase, but without lipids...
Not in yeast, but I think it's a good question. Maybe in other organisms it's possible that there are things that can mimic lipids.
Thank you.
Okay, thank you.
Last question.
Hi, thanks for the nice talk. A very basic question. When you see the RAD51 spot...
It's a RAD52.
Oh, RAD52, sorry.
Because we also do RAD51 and it's quite different.
Right. So when you see the spot, how do you know it's at the break?
Actually, I didn't explain it here, but I've been looking at RAD52 for many years with a LACO array close to it, and we already know from many experiments, even before I started on this subject, that when we induce a break with IC1, we can also have a LACO array next to it with a fluorescence RFP on it, and the RAD52 spot it's really on top of this LACO signal. So we don't have any hesitation on that. It's true that sometimes we have spontaneous RAD52 foci, so it's not excluded that among 100 foci that I analyze, 1 or 2 are spontaneous foci, but it's really a low percentage.
Okay, thanks.
Thank you again, Judith, for this awesome presentation. We'll pass to the next one. So hello again. Allow me to introduce our next speaker, Sébastien Huet, Assistant Professor in the Institute of Genetics and Development at the University of Rennes. His research project focuses on the study of chromatin remodeling observed during the early stage of DNA repair mechanism. Today, we will explain how to investigate reaction diffusion dynamics of protein in the nucleus of living cells using a fluorescence-based method. The floor is yours.
Okay, thanks a lot for the intro, and thanks to the organizers to give me the opportunity to present our work. So as you can guess from the intro and the title, basically I will not really talk about what we do currently in research. I will just have a little part at the end really focusing on the work of the lab, because I wanted also to more generally talk about reaction diffusion and how we can assess it in a living cells. And then I will give an example which is more related to DNA repair at the end. So basically just some basic definition to start with. So what do we call reaction diffusion? So basically reaction diffusion, as a very simple definition, is something that reacts and diffuse. So basically a bit like everything in a cell in a way. But if we go a bit more specifically, then what we are going to consider is basically a protein of interest that we want to analyze its dynamics in the cell. And basically this can exist either as a free state, so a state that can diffuse with a diffusion coefficient that we can define as D. And it can also bind to some partners or some structures. Let's say some structures, it makes things easier, which are relatively immobile, at least at the time scale we are looking at. So basically you have these two free and bound, and there is a kind of an exchange between these two populations, and we can basically model this with this kind of second order equation that we will even simplify for the rest of the talk, where we basically assume that there is this kind of first order with just a k-on and a k-off. So this is obviously very simplified, but at least I tried to show you that we can already do quite a bit of things by analyzing this kind of kinetics. So why do we care about that? Because basically, I mean, this kind of dynamics, which are very general, as you can imagine, is basically everywhere in the cell. So you have lots of proteins that would keep constantly moving and binding. So getting a precise idea and quantitative idea, which is obviously often the problem, is crucial to understand how these guys are going to go through the cell, to move through the cell. And just to give you an example, which is a bit closer to what we're interested in. So, for example, let's imagine this protein, so or GG1, which is a protein that is in charge of clearing these lesions in the DNA, which are called 8-oxo-G, so an oxidative state of guanine. Basically, this guy is very deep in the DNA. It's hardly accessible from outside. So to find it and to cleave it, basically, OGG1 needs to let's say scan the DNA and scan the entire nucleus to find the lesions and then remove it eventually. And what we don't really know is actually what's the mechanism, or what the let's say the time scales that limits this process. Is just basically finding the lesion that takes time, or is it cleaving it? So, and as you can imagine, these reaction diffusion processes that I mentioned should allow to basically address this kind of questions. So, okay, so these are the bases. Now let's see how we could assess that. So there are actually many methods that would allow to do this. I will just focus on two. One is FRAP. So I guess you all know the principle of this. So here I just mentioned FRAP as a general method, like photoperturbation, which involves either photobleaching, activation, or conversion. And the idea is to locally modify the steady state distribution of molecules and see how they recover to a new steady state. And the other approach that already was introduced yesterday and also several times actually during the week is this fluorescence cohesion spectroscopy. So here the idea is again to look at the dynamics of the protein, but in a different manner. Here you do not do pictures anymore. You just focus your laser at a point of interest and you look at molecules, so again in fluorescence, that are passing through the volume. And when these molecules are entering and leaving the spot, basically they do this kind of fluctuations in the detector, and by autocorrelating these fluctuations, then you get an autocorrelation curve that actually tells you how dynamic this protein is within the volume by looking at how fast the decay of the autocorrelation is. So basically, with these two approaches, you can get an idea of how dynamic or not dynamic the protein is. So before going into a real analysis of this data, then just some very basic stuff. So when you have FRAP data, then the first step is to pre-process them to end up into something where you correct it for background and bleaching, and you end up into this kind of curves eventually. Just a little reminder, and this I think is important to keep in mind, is that when you do FRAP, you assume that when you get a change in the recovery, it comes from movement of molecules, but you need to remember that there are other processes that could actually lead to change in fluorescence intensity. One of is this kind of reversible photoperturbation or reversible photobleaching, for example, which is that even for a protein that would be purely immobile, after FRAP, you would get a bit of recovery coming from the fact that some of these proteins that you bleached are actually not bleached in an irreversible manner. And the same holds true for FCS. So if you look at the correlation curve, then you would mainly assume that the fluctuation you're seeing are actually related to movement, but this is actually not true, unfortunately. And you have many other processes that could create fluctuations, and these are going to appear on the FCS curves, obviously, like this triplet or other processes. So this is something to remember if we start to want to be quantitative, that basically movement is not everything in our data, and we will have to play with this. Okay, so you have your FRAP curve, you have your correlation curve. So the first thing you can do, and this is what many people actually are doing, is first get a kind of a qualitative analysis of this data. So you can get this, from this recovery, you get a kind of a mobile and immobile fraction of molecules. You can get also a characteristic time at which the mobile fraction is recovering. Also from this FCS curve, you can get a residence time, which would correspond to how much time the molecule is spending in the focal volume. So all this gives you a kind of a qualitative idea of what's going on and how dynamic the protein is. Unfortunately, this is purely, let's say, qualitative, and it doesn't really tell you why this protein is more or less dynamic, which kind of processes this is underlying. Is it pure diffusion? Is it diffusion plus reaction? Is it reaction only? So to go further, then you need to start to build models, and this is what I want to go through now. And if you go through this, basically, if you go under the simple model I explained, then you end up into trying to describe your system with two main equations. One about the population that is diffusing, that will follow this kind of differential equation in space and time, and the bound fraction, which will actually be associated with the second equation. And if you look within these two equations, then you see that there is one term related to diffusion, only for the free fraction, because this is the one that can diffuse, and a reaction term that you find in the two equations. So this is what we have. So now the idea is that if we have a set of two equations, then we should be able to solve them. And by solving them, then we should be able to find the free and bound expressions in space and time. But as you can imagine, for diffusion, let's say differential equations in space and time, this is not an easy bit. So you need to simplify a bit the system, and I will show you now that there are actually simple regimes where you can start to play with. So a first simple regime that you can look at is a regime where basically you can assume that we are in a reaction limited situation. So what does this mean? So let's take the example of a FRAP experiment. So I bleach within a given area, so these molecules turn to black. Basically, if you are in a reaction limited regime, then you assume that what takes time in your recovery process is a reaction, so the binding. So basically, another way to say that is that the lifetime of the bound state is much longer than the diffusion time between two binding sites, which means that basically, if you start from the initial point just post bleaching, then these molecules, they will have the time to diffuse all over the bleaching area before starting to rebind to these binding states, which are these little squares here. So basically, you can assume that diffusion can be infinitely fast in this regime. And then if you see what's happening regarding the equations, it means that you can forget about this term, because basically this is always at 0, because diffusion is infinitely fast. So you end up into just these reaction terms. And this actually can be solved relatively easily, because then you end up into, if I focus on the FRAP data, some recovery curve where you have some kind of instantaneous recovery due to diffusion and then some binding. And from this, you can extract information about the k-on and the k-offs. So with this, in this regime, then you start to be able to extract some meaningful parameters. And there, the opposite scenario, here in the opposite scenario, then the lifetime of the bound state is now much shorter than the diffusion between two binding sites, which means that basically the reaction, the binding reaction, can be considered as always at equilibrium. It's infinitely fast, let's say. Which means that if now we go to these equations, it means that this term is always equal to 0. Basically, this reaction is always at equilibrium, and it gets rid also of this term. So it seems like you only have diffusion. So it's a process where basically if you look at the curve, you get a recovery that looks exactly like diffusion, except that the diffusion coefficient you measure is not a real or an absolute diffusion coefficient. It's an effective one, which is actually depending on the real diffusion coefficient, so when you're free, and also the ratio between k-on and k-off. So based on this kind of, or within this kind of regime, basically the only thing you can extract is not a k-on and a k-off, but just a ratio between these two reaction rates. So this is the different extreme scenarios where we can simplify enough the system to get some analytical expression that now we can use to fit our data. So this is true for FRAP, but this is also true for FCS, where you can basically get analytical expressions for the two different scenarios. So okay, good. We have two models. We should be able to fit now the data. So classical approach, I try different models, and I choose the best model. Unfortunately, in many cases, actually, because of the noise on the curves, it's often difficult to distinguish between these two models. So just by having these curves, it's often difficult to say, okay, we are rather in a reaction limited or in a diffusion limited situation. And also, these models that we built, I mean, I didn't really say that, but they often assume different things, like, for example, the diffusion occurs only in 2D, that the space around the spot that you bleach, for example, is infinite, etc., which assumption may not be always valid. So in the end, it might be difficult, even with this model, to distinguish between the different situations. So you need to go further. Basically, you need to add extra information to your data to be able to choose for one model rather than the other. And actually, if you think about FRAP, you have extra data, and these extra data are the information about space, because so far, with this recovery curve, you are only interested into time. But you have space information, and this space information will be actually very useful. Because, for example, if I do, let's say, photoactivation of alpha nucleus of different proteins, just by looking at how the recovery occurs, I could probably say what, in which situation diffusion matters, or in which situation actually I am in a reaction dominant scenario. And actually you can guess already from these images that in one situation what you see is that this gradient you created by photoperturbing is actually progressively smoothing with time. And this is actually a sign of diffusion, because diffusion progresses in space. So basically, if you have this smoothing process, that it means that you need to take into account diffusion. On the other side, like here, for example, if you look at this, like this step here, then you see that there is no smoothing of the step, which means that here in such scenario, only reaction matters and diffusion can be disregarded for this scenario. Another way to look at that is to check whether your recovery curves depend on the size of the ROI, and basically larger ROI could either lead to a slower recovery In this case, space matters, which means that diffusion matters. And in other situations, whether you are with a small or a large foci or ROI for bleaching or photoperturbation, you get exactly the same recovery, which would mean in this situation that diffusion, again, can be disregarded. So by adding info, now I can really separate between these two models, and in the end, you may end up into some kind of scheme like this, in which if you look at the dependence to space of the acquisition you're looking at, then you can decide between reaction limited or a process that involves diffusion. And then if now you fit your data with a simple diffusion model, eventually distinguish between this simplified model, in which I only assume diffusion, and a kind of a mixed, where basically I need to take into account both reaction and diffusion. But this is, again, probably a simplified situation where, in particular, you can do this fit properly, which is not always the case. So if you want to go further, then you can also start to do spatial topol fitting. And now, just for the few next slides, I will move from the nucleus, so my favorite place, to the membrane, which used to be my favorite place a long time ago, where basically you can look at what's going on, but by fitting the curves not only in space, not only in time, sorry, but only in space. And what you can imagine relatively simply is that if you locally bleach a molecule that would be at the plasma membrane, basically if there is diffusion, then you would get this kind of enlargement of the spot you created, and because of the molecules that are redistributing along the membrane. But now if you just have binding and binding, then the shape of this curve should remain the same in terms of radius, but the amplitude or the amplitude of the curve should decrease. So basically, by looking at how the shape of the bleaching area is evolving, then you should be able to separate diffusion and reaction. And just to give you an experimental example, then we bleached this protein locally, and we looked at the recovery, and we fitted the data a long time, and we could see that we have a progressive enlargement of this area that allowed to give us some diffusion coefficient for the protein within the plasma membrane. So with this, then you can access these two basically scenarios. But obviously, this is only valid for simple geometries, basically assuming that you have just a local frap occurring, and that you basically can consider that all the membranes here is infinitely large, which is not always the case. So what happens if you are in more complex scenarios where you cannot make these assumptions? Then you need to go into a different strategy. And this different strategy is to go back to these equations and now say, okay, I don't do any assumption on the size of the area I want to bleach, on what's going on. I will really try to model as closest as possible the recovery process. And for this, then you go from an analytical approach, so try to extract some equations, to a numerical approach where you will try to simulate actually what's going on during the recovery process. And by to do that, so you discretize your equation in space and time. So you end up into some kind of equations like this. And this you can eventually solve using different numerical solver. So this is good because then you don't need to do any approximation or assumptions about the FREP ARI. The bad thing is that you need to code this first. It's just not only an equation. And you also, if you go into 3D models, you may end up into some relatively long calculations. But the nice thing is that you can really now basically simulate any kind of scenario in terms of FRAP, for example, where basically here the example is photoactivation of alpha of the nucleus. So this is the experiment, this is a simulation, and we can really fit the data at different localization within the nucleus with this kind of model, and eventually extract some quantitative information from this analysis. So it works pretty nicely, and in the end, you're really open to any kind of configuration. Just a little warning again, but this is, I guess, for fitting people, it's not really a surprise. A fitting model will most of the time give you a value, but to really make sure that this value is correct, and you need to explore relatively exhaustively the parameter space, because I mean, as an example here, the model will tell me that the residency time, so this inverse of the k-off is 0.5 second, which is the fit point here, but actually you realize that you have a very large uncertainty in your k-off, and actually the only thing you can say is that the k-off is higher than a given value, which would be here. So something to keep in mind if you start to do some fitting. Also, just a little thing about this 2D approximation. So basically, in many of these situations for practical let's say problems, you would rather stick to 2D. First, because I mean, when you do FRAP, you most of the time do FRAP in just a single Z-plane, and also because 3D models are often difficult to implement. The question is, how much my 2D approximation is valid? So basically what you can assume is that your approximation, the 2D approximation, is going to be valid if basically the system is invariant along the z-axis. So if I take the nucleus here as a pancake and I look at it from the side, then basically if I do the photoperturbation, then it should be all along the z-axis, obviously, because then you can consider that you're invariant. And if you're not in such situation, then obviously your fitting is going to give you some values that are not completely correct, and this is just a simulation we did at the time to look at what would be the impact of, for example, having a photo perturbation spot that would be smaller and smaller, so going from red to blue. So the nucleus is simulated as a square, so we look at it from the zx side, and here these two lines correspond to the sectioning we do with the confocal. And what you can see is that the smaller this spot, here these curves are progressively moving in this direction, and do that basically you overestimate progressively your free pool, and ultimately you can get overestimation compared to the ground truth that can be a factor of 2. So something to keep in mind. Luckily, I mean, at least for people working in cells, basically if you look at the kind of perturbation you do, so by looking in a cell, basically this assumption that you are, let's say, in a space invariant in Z is usually relatively correct. So you can probably, in most of the time, assume that 2D models are going to be fine, but still something to keep in mind. So with this, I would like to give you a little brief example of an application of this, and this brings me to the stuff we do in the lab now. So basically, we are interested, and Judith shortly introduced that just before. We are interested into this first step of the DNA repair process, and in particular, what I'm going to talk about is really the initial steps. So basically, when you induce a break, or when a break is created somewhere in the nucleus, then you need to face a first problem, which is to access this break. Because, I mean, this chromatin has been packed so densely in the nucleus, then the first problem is to access the lesion. So most of the time, one of the first steps that are occurring are actually local chromatin remodeling events, which, let's say, role is supposed to improve the access to the lesion. The thing is that this was a model, and we wanted to go further on proving it. So we tried to look at different aspects. And the first thing we did is to look directly at the chromatin remodeling process. And how we do that, we set up a relatively basic assay. So the idea is to use a photoconvertible protein fused to an histone, so H2B P-FP. So this protein is black and turns into red upon 4 or 5 activation. So this would be the position of the nucleus before activation, so it's obviously black. And then you irradiate at 4 or 5 these cells, and by irradiating these cells, you immediately photoconvert the H2B, but also induce damage. So basically, this line you create is actually a readout or a highlight of the damage region, and then you can see what's happening with time with this damage area. And something you may see and appreciate here is that the size of this region, the thickness of this line is progressively growing. And we interpret this as a chromatin relaxation process. So chromatin is locally opening, and I don't have time to show you extra data to prove that, but it's clear that this region is progressively becoming more relaxed, potentially by improving access to it. So we can quantify these processes, and also to cut a long story short, we also showed that this remodeling process, so this chromatin relaxation, fully depends on a protein called PARP1 that basically is coming at a very early stage on the damage, probably the first one to come, to recognize the damage and to bind to it. And actually this protein is creating some modifications, so ADP ribose chains, on histones and also on itself. And we showed that the creation of these ADP ribose chains or PAR chains actually inducing the relaxation, because if you block them, then you completely block the relaxation process. So at this stage we wondered, okay, there is this relaxation occurring at very early stage. This is within the first second post-damage. Could this actually trigger the access for DNA? And basically, so the idea was to test whether in a close conformation repair factor would be stuck or would not have access, and upon damage induction, because of PARP1 coming and relaxing, would a repair factor would now be allowed to come. So the way to look at that was not to use directly repair factors, but just some kind of sensors. So we use two types of sensors, some that would bind to histones, and some that would rather bind to DNA. And what we found relatively strikingly is that any DNA binding that would actually be expressed in the cell would actually somehow recruit to the damage. You see this line here, and this would be PARP dependent, which would mean that if we block the relaxation mechanism, then we somehow impair the recruitment. So this suggests that basically, upon relaxation, you have an increased access to DNA, but not to histones, which is relatively interesting, even though we don't understand how you can do that. But the clear thing is that might be easier to get access. But to really show this, you need to really understand what's going on, because, I mean, these sensors that we use, they constantly bind and unbind to DNA. So they move in the nucleus with this reaction diffusion model I just mentioned. And actually, if you look here, what could happen, basically an accumulation of the protein at the break could actually mean two different things. Either it could mean that your K-on is increased, so you come faster because of facilitated access, or you stay longer on the damage, so a decreased K-off. And this obviously has very different meaning in terms of biology. So we try to assess this K-on and the K-off directly at the damage to be able to distinguish between these two approaches. And we did that by fluorescence correlation spectroscopy. So basically, we combine laser photomanipulation and FCS at the damage, so within this line here. And we measured, with the model I mentioned before, K-on and K-offs within this area, before damage and post-damage. And what we found is that basically what happens post-damage, that you have a major increase in the K-on, while the K-off is also increasing, while for recruitment you would rather expect a decrease. So basically we suppose from this data that basically the recruitment is related to an increase in the K-on. And also, most interestingly, when you put PARP inhibitors that are blocking this ADP ribosylation and then blocking the relaxation, then you completely suppress or mostly suppress this increase of the K-on. So this actually suggests a model in which basically upon damage, then PARP1 is coming, pyrrelating its targets, and this leads to an opening of the structure that would promote the recruitment of early factors. And we now have shown that actually several repair factors are actually coming through this mechanism of, let's say, increased access locally, because we have this list that is not exhaustive, of protein that are known to be involved, or that we found to be involved in the repair, that are coming via this mechanism. So with this, I would like to thank the people that participated to this work, so the past and present members in my lab, the EMRIC facility, the member of the EMRIC facility, and also past and present collaborators. And I also thank you for your attention. I am ready to take questions.
Thank you again, Sebastian, for the presentation. We'll pass to questions, please.
So that was a wonderful talk. I'm curious now, we use 405 and a lot of different things to activate stuff. Are we also inducing a lot of breaks, or are the intensities that you showed very different than what we use for
Yeah, so no, you can be reassured. So what we do is that we do this with 405 with cells that are pre-sensitized to actually induce damage. Nevertheless, for some repair factors, even without ERGs, so even without sensitizers, we do see recruitment. So it's, 4-5 probably induce some kind of damage. Yes.
Thank you.
Thanks. So the finite element approach to fit the FRAP recovery, can you use this also to include some spatial heterogeneity in the properties for the recovery, like we saw nucleoli in the nucleus?
Yes, yeah. So that's the idea, that if you base yourself on the steady state scenario where you have the distribution at steady state, then you can access actually to the, let's say, the heterogeneity of the kaon, which is a kaon that you consider as homogeneous multiplied by the concentration of binding sites, and then you can get access to eventually the heterogeneity, yes.
Okay.
Or you can include it in the model at least.
And same for the diffusion?
Yeah, well, I mean, yes, it starts to be untrackable, I think, but in theory you can do that.
Thanks.
I was just wondering, for the first part of the talk, when you presented your analysis with your equations, have you tried to go into more complex dynamics? For example, when you have transcription factor, they can not only bind, but they can also, let's say, go to the chromatin, go in 1D, then have interaction in 1D. And so usually it's quite complicated, I know, but what do you think? Have you tried to think in that direction?
Yeah, so we try to make the system more complex, but I should say we would be already happy just with a simple model to have something that is actually working and that we can basically understand. So already I think by going more simple and already extracting meaningful information for the simple model, we would be able to say quite a few things. Going further, for example, looking at scanning along the DNA would be great. I'm not sure that within a living cells we can do this easily, or we would need to go single molecule eventually, even though this might not be also so easy. But here the idea would be maybe to go back and forth between in vitro and in vivo. And for example, in vitro, you can probably more easily look at scanning along the DNA and stuff, and maybe get, for example, some characteristic transit time along the DNA and check whether with the K-off you measure with the simple model whether this is matching or not. I would rather favor actually this situation going back and forth between in vitro and in cellular rather than trying to fit a super complex model with basically very noisy data.
Thanks.
A very nice talk. I have a question about the biology model. Like I understood that there is this factor which is arriving, is changing the chromatin, is modifying chromatin, and then it opens, and then using the other factor arrive. How can you establish this link of causation? Like, couldn't it be that it opens because other factor arrive later? And did you try to do the experiment without other factors? I don't know.
Yeah, so these, so for basically the sensors we tried, these are stuff that are unrelated to repair. So there is no reason why they would recruit, except because the DNA would be more accessible, basically. And we tried also, for example, we make sure that they don't bind to these chains, for example, that I mentioned. So the only explanation we find, I mean, it's always difficult to show that something is not happening, but is that it's an increase in access. And for these real repair factors, then we could, for example, remove the DNA binding domains, which are known to be DNA binding, and see that you completely kill the recruitment. So there are always several lines of evidence that it's this process that is actually involved.
Thank you.
Any other questions? No? Thank you, okay, Sebastian, for this awesome presentation.
Thank you.
I'm sorry. Another question. Thank you.
Just an announcement. This is to say that the program is a little bit changed, and we're supposed to get back here at 3:45 because or 3:50 because the students will normally give talks. Okay, thank you. No, it's changed. Okay, sorry, it changed every 10 minutes. So. Okay. So at 3 in the morning, next night, there will be the poster, okay?
So we get back here at 4:10 in the end, right? Yes. Hop. Tiens, hop, je te laisse. Je sais pas si tu peux
Ça doit être.
Ah.
Quoi?
Bah je sais pas qui c'est. Ouais. Non, bah laisse-le là, ça doit être. François? C'est à toi? Je sais pas. Il est là, il est là, ouais. Si tu veux te servir du tien, juste laisse celui-là.
Ouais, je veux bien, mais il me faudrait le tutuc.
Ah.
Parce que sans ça sert pas à grand chose. Hein. Oui. Ce truc-là, c'est à vous? Si. Est-ce que ça, c'est à vous?
Oui, c'est à nous, mais je sais pas si
Ah, yes, excellent. On va essayer.
Je suis pas sûr que il y a un zoom là. Il y a un zoom sur votre son.
C'est que des diapos, mais c'est moi qui présente.
Il n'y a aucun zoom, il n'y a pas d'intervenant en zoom, en vidéoconférence.
Peut-être après, mais c'est pas sur ma machine.
C'est pas sur vous.
Enfin, je sais pas.
Donc il n'y a aucun son qui doit être transmis dans la salle.
Antoine, tu as le. Après moi, c'est en zoom. D'accord.
Donc pas de son qui sort du PC.
Donc on va vous dire si le signal sort, s'il sort là et puis ça va après toi c'est
Tu peux regarder les voix et on va faire un courant qui sort. Et alors après il y a toute une statistique pour les analyser. Par exemple, quand tu as des particules qui tu veux dire moi je sais un petit peu en particules, par exemple ne rentre pas dans ce domaine. Et qu'est-ce que tu peu extrême, tu sélectionnes cette population. En fait, quand tu conditionnes ton processus par ça, tu obtiens des choses. C'est pas vraiment la condition directe, c'est autre chose. Parce que tu corrèles avec la géométrie.
Mais tu posais un verre d'eau, il y avait des
ça devient un truc subtil, c'est vachement intéressant parce que tu as une sous-population, tu l'as sous des
je suis pas particulièrement prêt, je pense qu'une fois dans le cours ça ira.
Ce que je te dis, c'est vachement intéressant.
Bon. C'est pas une très bonne idée. Enfin bon, non mais en même temps c'était sympa pour les jeunes de pouvoir se balader, mais donc ça m'a un peu... je suis un peu... Voilà. Ok, très bien, bah, écoute, ça a une bonne gueule. Moi je vais rester ici encore 5 minutes pour vérifier que tout tourne.
On peut faire, une fois que tu as fini, mets-le en plein écran et mets-le sur ta slide de
départ.
titre.
Ouais.
Puis moi, quand je fais la petite annonce,
Très bien, tu reviendras en arrière. Ok, très bien. Et il faudrait probablement que j'aille chercher un chargeur. En fait, je vais plutôt utiliser... Ils en avaient un deuxième, non, comme ça. Parce que celui-ci ne fonctionne pas très bien. Il faut appuyer 4 fois pour avoir le truc. Donc je vais peut-être éviter. Excellent.
Tu veux prendre ton chargeur?
Ouais, volontiers. Merci. Ok. Excellent. Mais t'es chouette, le talk de UE était très bien.
Ouais. C'est bien qu'il y ait pas que du SPT, qu'il y ait du frappe et du UCL.
Ça va me prendre un peu de temps, je vais le mettre un peu. Jusque là, c'était 40 secondes, je vais pas tenir. Déjà les 20 secondes. Ok. Parfait. Très bien. Et le pointeur, oui, j'ai pas vérifié. Je vais revérifier et puis ensuite je devrais... Voilà. Toc. Non, rien, c'est là.
Ah bon, tu as changé le petit truc.
J'ai changé le petit truc, mais il y a peut-être un bouton on-off que j'ai pas activé. Ouais. Bien. Interrompu. Hop. Sans doute pas la peine. Ok, alors, bah parfait, merci beaucoup. Moi je vais juste vérifier que toutes mes diapos tournent et ensuite je vous rejoins dehors. C'est 4h10, hein, c'est ça? 4h10. Ok. bien. Oui, bonjour. D'accord. Et que l'un, ils sont tous les trois.
Je vous en mets plein les yeux, mais je vous en mets plein de trucs, donc c'est pas bon. Vous, en fait, vous avez tout le haut nickel, et là, c'est obligatoirement spécifiquement de lumière, sinon je ne vous vois pas du tout. Et c'est-à-dire sur le direct, je ne vois plus, sur l'enregistrement pour la vitamine éternelle, je ne vous vois pas. Et je ne peux pas ne pas vous voir du tout.
D'accord.
C'est tous les intervenants, il faut qu'on voit que leur tête quand même.
Ouais, ouais.
Donc voilà, regardez, c'est pour ça que je veux dire, c'est que notez que j'ai demandé à l'ingénieur au travail de passé, il n'y a pas de note. Donc il y a quand même ça. Alors je peux, s'il y a 2 ou 3 diapos.
Donc il y a une diapo où je vous demanderai, vers la fin il y aura des diapos où je vous demanderai, je vous ferai signe et puis
vous me dites le numéro, vous avez une idée du numéro?
Oui, je vais juste passer mes diapos en ordre et je vous dis lesquelles. Je vous dirai laquelle c'est à la fin.
Je baisse à ce moment-là et je remets.
Tout à fait. Donc là, je pense pas qu'il y en ait besoin. Je vois qu'il n'y a pas de souci. Très bien. Ok. Il n'y a pas de souci non plus. Ok. Là, c'est pas trop fort. Ok. Très bien. Là, ça va. Là, ça va aussi. Ça va aussi. Ça va aussi. Bon, ça se voit très bien.
Sachant qu'on est bien d'accord sur le direct que je crée, sur la vidéo que je crée, c'est votre diapositive qui sort.
Oui, oui, bien sûr, j'entends.
C'est vraiment ici maintenant.
Donc c'est, là c'est celui-ci.
Ok, celui-là, il faudrait qu'on joue un jeu de lumière sur celui-là. Vous hésitez pas, je me le note, parce que c'est le petit jeune homme de l'enfant.
D'accord, je vous ferai un signe, d'accord.
Voilà.
Ok. Il y a surtout celui qui pose le plus de problèmes en fait. Ça va, je pense, pas de souci. Celui-ci, il faut que ce soit noir complet. Enfin, vous éteignez tout. Moi, je parlerai dans le vide, il y aura 30 secondes, il ne nous verra pas, mais c'est pas très grave. Et c'est juste cette diapo-là où vraiment, parce que là, on perd la moitié de l'information. Si vous voulez, il y a plein de rouge. On ne peut pas voir.
Cimetière, celui-là, et quand vous faites le signe.
D'accord.
Et comme le jeune homme, c'est un jeune homme qui est à la et c'est lui 2-3 secondes.
D'accord. Et donc, il y a cette diapo-là et la suivante, qui est en fait un crop du même film.
Ok.
Et ensuite, je termine. Donc, voilà.
Cimetière, c'est les 2 dernières avant la fin.
Les 2 dernières avant la fin, exactement.
Okay. Vous faites le petit signe de main.
Ok, je vous ferai un signe et vous me l'éteignez à ce moment-là.
Ok, c'est ça.
Super. Et je vous referai signe pour rallumer, hein. Il n'y a pas de... De toute
façon, après, il y en a 3 importants.
D'accord, oui, exactement. Ok. Très bien. Ok, parfait. Okay.
First of all, the mic, you have to see the green light.
Green light.
It's okay.
Okay.
And when you speak, the mic on the.
Okay.
Okay.
All right.
And
I'm
you.
pretty loud in any case, so it's all right.
It's okay. Then connect your computer. You have the HDMI cable here to plug on your.
Ah, okay,
computer.
okay, okay.
This one.
Ah, perfect. Okay, and then what do I do on that one when that one is on? You have a laser pointer.
Yes.
I don't know if you have.
I have.
Ah, you have the.
I have.
That's good.
Ta-da!
Perfect.
Perfect. Let's see. Does the movies work? Oh. Oh, so you can do it with that one? Yeah. Oh, okay. But I guess the lights are going to be off.
Yes.
So it'll be okay.
The light will be off.
Perfect. Amazing. Okay. Very good.
Perfect, sir.
Perfect. So we just so this. Ah, you put so you put this one in and that one.
Yes.
Okay.
Okay.
And
So
this
keep
one.
everything.
Ah, this one I keep. Okay. Perfect. And then the mic, I just pick it up down there.
Yes.
All right.
And you put it behind your mouth.
Okay. I'm trying to think. So. Okay. I have. I'll just bring this up for water. Oh, you have water bottles?
Again with nothing.
No, it was boring.
Wow.
I think it all dissipated around 4:30.
Yeah, probably. So did keep you up?
No.
Okay.
I thought it was great.
Were you close to the?
No, it's by the water.
By the water.
I think I think it was great. I was like, it's a good sound. You know?
Yeah.
I was talking to who's the co-organizer from Grenoble who's retiring. Yeah. Steve Lundford. He's one of the founding members of MIFOBIO.
Right.
Oh, sure. Yeah. Anyway, he was telling me how in the early days they had their party the first night, and then students had to come with presentations.
All right.
Some like 8:30 a.m. the next day. He's like, they only did that once. He's like, so quiet try. Exactly. It's like nobody was really asking questions. The people who were presenting. Yeah, because I mean, this is like genius. So they had the party last night, and then he started. And you choose to sleep. That's fine. I mean, I think some of them went to bed at like 5. You know.
Yeah, bro.
This morning at lunch, I had breakfast. It was sit on the ground.
It's good to not to start
Yeah, that's
many
true.
months.
No, I think he sort of puts it because it's a nice opportunity to meet and kind of connect. And then just to make their own party up a lot.
It's all more
It's one of those. This one is dead. No, it works. It works. Yeah.
I think that's the point.
I'll use that one.
Session.
There. Yep. Nobody told me. I was like, unless the lamp is on fire. No.
Yeah.
You can't do it. Close is fire, so this is great. But then it says you can do a pile of stuff.
When you look at home, you cannot escape.
Yeah.
That's stuff. But then either you actually escape and you come back and you have to find it.
Yeah, right, exactly. That's the thing. You pay the price. Pay the price, right, exactly.
It's like when you take vacation when other people don't.
Yeah.
Think that you pay.
Bon, ça va.
Il est que 13, hein. Okay, welcome everyone. So before we get started with the session, I just want to make two announcements. First, so that's the last session of module 6 about molecule dynamics inside living cells. And so actually this evening we're going to have the roundtable. So anyone who's interested in this topic and wants to share thoughts and discuss where the field is going, please join us. It's going to be at 9:30 in the lounge. So I think it's just above here around the pool table. Yes? Okay, yeah. Well, let's meet up there, and if it's not there, we're gonna move. And the second thing I would like to advertise, actually, so there's going to be a symposium in the memory of Maxime Daon, who I guess many of you used to know. Maxime passed away a bit more than 3 years ago, and this is the second symposium in his honor, in his memory, with really great speakers from all over the world, many of which actually work with Maxime or actually followed up on his former work. And so this is organized by Bassam Haj, Mathieu Copé, and Vincent Studer. And I invite you to register if you're interested. You can come on site at Institut Curie, but it's also an online symposium that you can follow online, so I strongly encourage you to participate. And with this, I'm just going to give you the microphone to the chair of the session.
So our next speaker today is Dr. Francois Robin. He's a PI from Biology Institute from Paris, Salle. He obtained his PhD from Developmental Biology Institute of Marseille, where he studied the cellular control of morphogenesis. And then he moved to the US and did his postdoc at University of Chicago, where he developed imaging tools to visualize the individual molecule at the cellular surface in living embryos. Starting from 2015, he leads a research group to study and engineer cell mechanics from the cellular to the tissue level. And let's welcome Dr. Robin.
Is it too loud? No. Okay. First, I would like to thank the organizers for inviting me. I've had a tremendous time here since I arrived. It's the first time I come to MIFOBIO. I'm learning an amazing amount of new things, so I'm really thrilled to be here. Thank you. And with this, I'll get started. I'm going to tell you two stories today, looking at the architecture of the cell contractile machinery. As the introduction mentioned, I am working at the Institut Biologie Paris-Seine. I'm fascinated by morphogenesis, which is what I started my career on. This is work I did during my PhD, looking at the shape of cells during the early embryonic development of a small marine invertebrate, Cyanitis nalis, and already looking at these very few first hours of the embryonic development of this marine animal, you can see that cells change shape, and you have motions of cells that displace the cells within the embryo to position them at the right place. And the organism I use today to study this is the nematode, C. elegans. And it's a really nice model system. First of all, it's very small, so the worm is 1 millimeter long, and it has eggs in it. It develops over 2.5 days, roughly speaking, so from an egg to the next generation egg, you have about 2.5 days. And it's a really powerful model system because it has a fixed cleavage pattern, so if I look at any of these cells, I can get the name, and it's always at the same position. And the second thing, it's extremely powerful genetically. It's very powerful genetically, but also very useful for microscopy. And what we're interested in this little beast is the machinery, the molecular machinery that drives cell shape changes. And this machinery is composed of actin filaments that you can see here, labeled with phalloidin. So this is the C. elegans egg. It's 50 microns in length and about 30 microns in width. And this actin network is assembled just beneath the plasma membrane, over a depth of about 200 to 500 nanometers beneath the plasma membrane. And the actin, together with the myosin minifilaments, which use ATP to generate forces, are capable of generating extremely dynamic behaviors, auto-assembled, auto-organized behaviors. So this is a 1-cell stage dividing to 2-cell stage and 3-cell stage and then 4-cell stage. And the myosin is in green and the actin is in red. And this machinery, composed of actin filaments and myosin motors, is actually able to generate a wide variety of cell behaviors, ranging from cell division to cell polarization and cell shape changes. And so this is the same movie, but this time we're playing it a little bit slower so that your eyes can actually see things. And now I hope you can appreciate that it's really nice. We can see little dots of myosin moving around and actin filaments that we can sort of distinguish, not really well. This kind of movie, we're requiring the dynamics of the actomyosin relies on the coordinated behavior of these actin filaments and these myosin minifilaments. But in muscle, we know very well how this machinery works. It's the same molecules, actin filaments and myosin motors. They're well organized, and the myosin, which is a myosin 2, tends to walk towards the plus end of the actin filaments, which is localized in the muscle here. And because these guys walk towards here, you have contraction. Okay, so it's very easy to understand how it works. In embryonic cells, the cortex that I showed you earlier, it looks more like this. So understanding how this mesh of stuff, this ball of noodles, is able to actually generate contraction is not completely clear. And one of the things that has emerged over the past 10 years is the fact that actomyosin actually contracts in pulses. So the first place where this has been observed has been the mesodome of Drosophila, where you can see that these cells invaginate. They form this sort of ridge here. They internalize, start to. And they do so by constricting apically, and this is driven by myosin. So myosin is localized at the place where you see this. But the myosin doesn't localize in a stable manner. Rather, it it passes. So in this movie, you can see the apical region of these cells, and you see that in purple, you have this phased accumulation of myosin that comes and goes. And if you measure the area of the cell, you see that it decreases, then stabilizes, and decreases and stabilizes. And so you can look at the derivative of that and see that you have phases of contraction. And a body of work has established that these post-contractions, which is what they termed, work by contracting the apical surface, which relaxes a little bit, and then contracting again and relaxing again. And so these post-contractions are extremely important to generate cell deformation, and they've been observed, as I've told you, in Drosophila. So this is a wider scale movie of the exact same process, where you can see the myosin accumulating and the cells shrinking. But they've also been observed in frogs, in worms where they drive polarization, and during gastrulation in worms where they also drive gastrulation. And also, they've also been observed in mouse embryos. And what we've decided to do was to use these pulse contractions that we could observe at the 2-cell stage to try and understand how these pulse contractions were working in a very simple system. And I hope you can see that you have, during these pulse contractions, accumulation of actin and myosin, and these are the two things I'm going to talk about today. And the questions that we had, looking at this ball of nodules, was, is it self-organized? How do you define the time scale of this? And how is it organized spatially? And I'm going to tell you a little bit about the spatial organization today and elements of information that we've gathered. And the way we visualize these guys is by using turf microscopy. So I've told you that the cortex is a very thin layer of material. It's 200 to 500 nanometers in thickness. And we use turf to, well, not exactly turf, high-low microscopy to visualize this thin layer at the cell surface. And one of the things that they did during my postdoc was to look at, develop ways to look at these molecules at the cell surface, because when you look at your movies, you have too much information actually, and it's very difficult to extract quantitative information from what's happening. So one of the ways that we look at single molecules is to reach this single molecule land, which is a nice place where you want to be if you want to not have too high concentration of stuff. And to do that, for example, we use GFP RNAi, and that allows us to get to a point where we, instead of seeing a full network of actin, we can just see dots. And when we look at the intensities of these dots are actually represent monomers and dimers, but if the density of your dots is low enough, then you only have monomers. And if you look at the intensity over time, you can see that most of the time you have single step for the bleaching, and sometimes you get a doublet with a first step and the second step for the bleaching. And looking at multiple of these molecules together, all fused with GFP, we can actually visualize, realize that the GFP intensity is the same for all of these guys. Okay. And what we want to do is use this type of analysis to extract, just to give you some sense of the control that we have over what's happening. We want to measure the dynamics of fixed binding and unbinding from the cell surface. And what we observed very early on was that if you look at your embryos with a very slow frame rate, so an image every so often, very rarely one every 5 seconds, the number of molecules that you observe is sort of constant. But if you turn on the laser and wait a bit, then you reach a second state, which is sort of steady. It's slowly decreasing, but fairly stable, which is in which, in this case, you have, in addition to a K on and K off, which is the dynamics, the steady state dynamics, you have added a photo bleaching component. And we can write all the equations for this, and it's actually very easy to extract, well, very easy, fairly easy, to extract the K on and the K off and the KPH for our system. And so if you turn the laser on, you see that your density of molecules decreases. You turn the laser off for 30 seconds, and your density is restored, because you're essentially renewing all the material that you had at the surface, but because you have a huge volume of which to take from, it doesn't change much your initial density. And if you now change the laser power progressively and increase the laser power, what you can see is that the inference that you have of the turnover rate doesn't change, but the inference that you have of the photo bleaching rate increases linearly. And so this is the type of movies that we had collected at the time, and here what you can see is actin labeled with with GFP, and the movie is accelerated 60 times because actin is sort of lazy, it doesn't do much, and so it's boring if you don't accelerate it much. But what you can see from there is that we can actually visualize molecules for a very long time at the cell surface. Okay, and this is quantification of this. For about 500 seconds, we can visualize single molecules at the cell surface, in the case of actin. So we are, today I'm going to tell you two stories. So this is published, and I want to tell you two stories. The first one is working on, based on this, exploring the functional architecture of pulses during pulse contractions. And the second one is to take a closer look at myosin in the cell. And so these two stories work on these two actors of the pulse contractions. So these pulse contractions that we see are actually driven by a Rho GTPase, which is a molecule that activates actin and myosin. And what I'm going to show you in the first story is what happens when we look at formin, the formin which is called Psych1 in C. elegans. And this is a story that was developed by Vlad Karstash, who might be around there, and who was a postdoc in my lab at the time. And formins are dimer molecules that sit at the barbed end of actin filaments and add monomers progressively. So essentially, what you have is a molecule of formin which adds sequentially, consecutively, monomers at the barbed end of the cell, of the actin filament. And now, if we look at these molecules of formins in cells, I'm going to ask you to look at this little dot, which is right here. We can see that they tend to move, okay? But not all of them. Some of them do move, others actually don't. They stay put in the same place. And to visualize that a little bit more easily, what I did was a maximum intensity projection, a very simple process, and what you can very nicely see here is that you have these two populations of formins, one that move ballistically in the cell, and a second population that sort of stays put. And this is actually very interesting because it tells us these formins, they sit at the barbed end of actin filaments. And so if they're moving, that means that they're adding molecules of actin at the barbed end. But if they stay put, it means that the actin is actually not polymerizing. And so looking at just the behavior in the cell, what we can do is have a dichotomy between two populations. One population of active formins that's actually elongating the actin filament, and a second population which is sort of sitting there and doing nothing. The second thing, which is very trivial, is that we can measure the speed at which these guys move, and they move at 1.2 micrometer per second. And that's interesting because when we see the formin moving, what we see is an enzyme catalyzing an enzymatic reaction. So we see an enzyme, and what we measure actually is its enzymatic rate, the rate at which this reaction of polymerization occurs. And so we have, roughly speaking, about 430 monomers per second added at the barbed end of the actin filaments. So the next step for us was to do a little bit of single molecule tracking, and to do that, we did the single molecule tracking, but we analyzed the tracks that we got. And to do that, what we did was simply to look at the mean square displacement as a function of time, and what we wanted to separate was these guys, the formins that move, from the guys that don't move, okay? And to do that, we just plotted the log MSD versus the log time, and we could nicely see that we had a distribution of populations. And now if we look at the distribution of slopes of these MST, we get this distribution, which is kind of nice because we have two peaks here. We have a first peak, which is clearly below 1, which means that the molecules are subdiffusive, or they're diffusive, but our resolution doesn't allow us to explore that. And we have this second population that peaks around here, which corresponds to our superdiffusive formins. So what we did was to just map this on this kind of movies, which is a little bit crowded, but in our case, we're interested not in tracking these guys for a long time, but mostly in informations about their orientation. And just from this observation of this form infused to at single molecule levels, we had two information. The first one is that we have these two populations of forming, one boring, the other one more interesting. But also, we could separate these guys and look at the sequence of recruitment at the cell surface. And what we saw was that there was, so might not be completely easy to see, but there is a slight delta T between the recruitment of the superdiffusive formins, the green ones, and the recruitment of the subdiffusive ones, in a way that was sort of surprising to us, and which is that the superdiffusive formins were coming first. The second thing that we observed was that these superdiffusive formins, at least the long ones, represent only 10% of the total population, and even when we compare them with the entire population of formins that were long-lived, they only represent 1/3 of the population. So that means that when you look at your formins in live embryos, about 2/3 of them are not indicative of actin polymerization. Okay, and now going back to what I was saying earlier, I've told you that these pulse contractions occur by local recruitment of Rho, and that means that locally you're recruiting these formins. So Rho lies just upstream of the formins and recruits the formins, and so you see flashes of regions where the formins flash, so where you see them accumulating. You have one here, you have another one here. And what we thought was, okay, maybe because they're recruited in an isotropic manner, not all in the same region, maybe they also assemble a network which is not isotropic. And so the architecture of the network would be actually modulated by where the formins are recruited. And this is actually important, and I'll explain why a little bit later. And to test that, what we did was to take one of these pulses that we can identify by eye, which are the regions where you have these congregations of formins, and look from within the pulse to outside the pulse whether we had a trend of direction. Are the formins in this region isotropic, and are the formins in this region anisotropic? And to do that, we just split all these regions and measured the angles of the formins going through the region at different points in times. And so formins going out of the region, so escaping the region where they're being recruited, would map here, and the formins going towards the center would map here. And so this is a lot of information. That's fine. There is only a small region that's of interest to us, and that's this region. When the pulse is initiated, so around here, this is the number of formins in the region. When the pulse is initiated, you can see that the distribution of angles becomes anisotropic with formins mostly going out. Okay, so that makes sense. What I'm telling you is that when you recruit formins in a region, they move very fast and they escape the region where they've been recruited. Okay. But from a biologist's point of view, that's actually very important, because so this is the process as we see it happening. So you're recruiting formins in the middle, and they escape, and the established network, which is biased with formins going out. And what that tells us is that we have barbed ends on the outside, and the barbed ends are the plus ends, the ones towards which the myosin wants to go. And so if you now recruit myosin in the central region, what the myosin wants to do is walk towards these barbed ends. And so essentially what you've done is generate a system in which these guys are going to try and pull these guys back in. So think of a spider in the middle of the web trying to reel the fibers of the web and contracting the actin network. And so essentially what we've done, just by tuning the local concentration of Rho, we established a chemical gradient. And the formins, what they do is actually convert that chemical gradient into a structural gradient. And because when these guys reach the border of the cell, what we imagine is happening is that the actin locks over there, and is able then to generate contraction and a shrinkage of the cell. And so what we've done is convert just a chemical gradient into a structural gradient. And so just looking at one actor in this cascade, we've been able to establish that there is an architecture disperses, and this architecture is actually prone to contraction by its design. But it's kind of awkward, because now this is what we observe in C. elegans with the plus end on the outside, the myosin that wants to walk towards this plus end, and as a result pulls the actin filaments back in. But if you go and look in yeast, you have the exact same ingredients, the myosin in red, the formins in yellow, and the actin filaments in green, but they're inverted now. So during the cell division in yeast, what you have is formation of what's called nodes. These contain all these three ingredients, and the model that has been suggested to generate contraction in this system is that the formins actually elongate actin filaments, so they push out the actin filaments. So instead of moving, now the formin stays put and pushes little actin filaments out. You have the plus end on the inside, the minus end on the outside, and that minus end will drift around and at some point find another node. And when it finds another node, it generates contraction. And it's actually, again, for a biologist, very interesting, because what you're doing is you have the exact same ingredients. The only difference between this guy and this guy is that this guy measures 4 to 7 micrometers, and this guy measures 50 micrometers. Okay? So you have one of the magnitude in terms of size, and actin filaments, they're not rigid bodies. You can bend them. And you could not imagine pushing an actin filament which is tens of microns long in a C. elegans cell. But you could imagine pushing this guy out in a yeast cell. The distance between the nodes is much more smaller. And so evolution with the exact same ingredients has generated two machineries with exactly opposite geometries to drive the same functional output. The second story has been developed by a postdoc in my lab, Shashi Kumar Suman, and it's looking at myosin. And myosin it's a huge molecule. So this is your typical myosin. It's a hexamer composed of 2 heavy chains, 2 regulatory layer chains, and 2 essential regular layer chains. And this myosin can assemble into mini filaments. And these mini filaments can be huge. They can be made up to hundreds of hexamers. And for 100 hexamers, you have a machine that weighs about 25 megadaltons. So that's a big baby. And what we did was to follow a strategy that was used before. I've shown you a movie of myosin earlier, and the movie had myosin tagged at the C-terminal, so around this region. And what we did was to just shift the tag on the other side, and that's something that had been done before. So this is the movie of the myosin tagged in the center, and you can maybe appreciate that these guys make little dots. And this is a movie of myosin that has been tagged on the outside. And maybe you can appreciate now that these guys make what looks more like rods. Okay? And so, looking at this, so this is a side-by-side comparison. This is the exact same molecule that is tagged. It's the same gene, right? But one is tagged on one end, and the other one is tagged on the other end of the molecule. So we have something that looks like a rod, and we thought, okay, maybe we can get something out of that. So we made movies in a situation where, so this is too high density to work, so we moved to something that was a little bit more compatible with tracking, and we did some tracking, and that looked like very bad, like you don't want to work with that. So the tracking was very good, but guessing the orientation from the shape of this guy doesn't work very well. And so the question that we had was, can we improve the spatial resolution of this object? And for this we were lucky. Our objective was to be able not to see a rod, but to see 2 little dots. And for this we were lucky because, well, Palm Storm, these guys move very fast, so it's difficult to imagine that we would be able to visualize them dynamically. with Palm Storm. Instead, in our hands, the embryos would not be happy. And again, we were very lucky because at that time we had a discussion with Emmanuel Bourpère and Pierre Mahou, and we realized that there was a sim turf just next to us. And so now this is our movie of mycin, labeled at the two hands, looking at turf. And I hope you can appreciate that now, instead of looking at a blob, we can see two little heads dynamically moving around. Okay, so we like to be sort of quantitative when we can. So one of the first things we did was to measure the distance between the two heads. So the way we designed that was by saying, okay, we're going to take a head, look at its closest neighbor, and then look at the closest neighbor of the closest neighbor, and if the two matched, then you have a doublet. Okay, so it's like find someone around you. If he's as close as you are from him, then you're good. And so we then measured the distribution of the distance between the doublets, and we got this. So this distribution is actually very peaky. It peaks at about 285 nanometers, which is consistent from what we know from the literature. And there is a distribution which is peaky, but which is skewed slightly more to the left. And it sort of makes sense, because if you look at an object in 2D, you can't make it longer than its length, but if you tilt it a little bit, you can make it shorter. So it might be that this skew in distribution comes from that. Okay, the second thing that we did was to use form in molecules and measure the intensity of single molecules in this strain and compare that to the intensity of the multimer that we had at the beginning. And when we did that, we got that the ratio of intensities were about 2800 to 150. That's about 20 mycins, more like 16 actually. 20 mycins per minifilament. And that was for us very interesting, because typically people would have thought that the number of heads was around 50 before we did this in C. elegans. And data from human cells would be more around 20 to 16. So we're actually very happy about this. But as a biologist, again, this is very interesting and quantitative, but what we really were interested in was new behaviors that we're now able to observe. And so I'm going to show you a few of those that we hadn't seen before. And this is actually very interesting, because when you unlock a resolution, you can see new things that you hadn't imagined before. And so this is one of the things that we started seeing, which is one of these minifilaments sort of grabbing two ends and then letting go and grabbing their other end. Usually it works better because I have two hands to do that. With the microphone it's a little bit more difficult. The second thing that we started seeing was sort of awkward, and it was periods between two bindings where the mycin would not be resolved. So in all the other mycin that you can see in this movie, we can see both heads at all times, but sometimes we would get guys like this one, where we actually don't see the head. It sort of moves too fast, and that was sort of striking to us. And the way we interpret this is that mycin can unbind. Okay, it has two hands to grab to the actin, but sometimes next one. Sometimes when it unbinds, it's just stuck in a meshwork of actin filaments. So imagine the mycin is this 300 nanometers long object, and it's within a network that has a pore size which can be, I don't know, 150 or 200. So what you have here is the signature of confined diffusion, which is your mycin is trapped in the network and unable to diffuse away. And eventually, what the picture that this paints is that mycin is a little bit like a children's net. It has two arms to grab the net, but even if it lets go of both arms, then it still won't fall in the cytoplasm because it's sort of trapped by the network. Okay. And now the last thing that we observed, and which is in my mind the most interesting one, was that we can actually observe, looking at the initial movie that I showed you, that mysin would tend to form long cables, and that was something that we had not seen. I mean, I've been looking at these movies for 10 years. Without this resolution, we had no idea. And if you look at these cables, you really can see, let your eyes look at that, sort of long cables of mysin that seem to be stuck around and all aligned along very long distances. And just taking one picture, you can actually visualize them. You don't need to go very far, and you can see that you have all these mysin here, which are perfectly aligned, all these guys here, all these guys here. So just from a picture, we did that. We extracted mysins that were all beautifully aligned. And then we measured, and then we looked at the dynamics, and this is one of these example dynamics. So looking at this guy, we can see the network closing. So I'm just going to show you that with a little bit of the drift. And now you can see the mysins closing in on one another. So if you make a chymograph of that, you get this. And we can measure that the many filaments are about 280 nanometers apart from each other. And what's really cool about this is that now we know that the actin cable on which this guy is sitting is bipolar, because mysin walks towards the plus end. So if all the plus end point in the same direction, you don't have contraction. To get this type of contraction, you need to actually have a mixture, a bundle which is a mixture of filaments in both orientations. Okay, and this is probably, so this is a movie of a two-cell stage embryo moving to three-cell stage. So this is the cleavage furrow, but this is not what I want to show you. I want to show you one of these cables forming. And so, this is an extract from the previous movie. If you look at this actin cable right there, what you can see is that when it's turning, it's being stretched. And as it is being stretched, it starts accumulating these mysins. If you look at his friend, which is just below, this guy, which starts with some mysin on it. I hope it's this guy. Yep. It sort of bends, and as it bends, all the mysin that was on it starts to fall off. And just, again, we can identify these long cables, and if we compare the stability of the mysin which is in the cables to mysin which is outside of one of these cables, we get a threefold increase in the turnover when you're outside of a cable. So when you're in a cable, you're extremely stable. And the reason I think this is interesting, because when you pull on a network of actin, it's sort of in every and all directions. But a few of the cables in the network are going to bear most of the load. So this is numerical simulations made by Taeyeon Kim a while ago now, where he pulls on an actin network and look at where the strain, the forces distributes. So in this network, you see that you have few fibers that actually bear the load of the stretch, which is imposed from the outside. And what we think is these fibers that accumulate the stress and line up, they actually will then recruit mysin. And what you have is actually a system in which you have a mechanism to maintain homeostasis, a mechanical homeostasis. If your network is stretched, some of the fibers will recruit more mysin and then fight off that stress. Okay, with this I will reach the second conclusion, which is that mysin exists as bipolar functional units in C. elegans, 280 nanometers long and with about 20 heads, and that these guys are bipolar, can display confined diffusion, and that you can visualize just by increasing slightly your resolution cables that support mechanical tension and can act as mechanical actuators to fight off and maintain homeostasis. And with this, I would like to spend a few seconds to thank the people who have been involved in the work. Camille Planck, who is our technician, Vlad Kostash, who did the work on formins, Shashi Kumar Suman, who is now in Strasbourg and who did the work on mysin, and our collaborators working on SIMTUF microscopy, Emmanuel Robert and Pierre Mahou, and all the people in our institute, as well as our funding bodies. Thank you for your attention.
We thank the speaker for the very nice talk. So now it's question time. Okay.
Hi. On a larger length scale in tissues, usually when cells get stretched at their cell interfaces, there is a recruitment of myosin 2. Do you think your data suggests that this kind of feedback could have as an origin a pure geometrical explanation, a little bit like you propose on your
it's
system?
Possible that, I'm not sure that I would say that this would be my first guess. It might contribute, and might be the first thing that started evolutionary, but I'm not sure that it would be. I mean, when you have these sort of stretches, you can also visualize rho very often, and so I'm not sure how the two mechanisms work together.
Okay.
Any other question?
So I guess you don't have a current understanding of how myosin aligns.
So what we think is the cables stretch and then myosin is recruited. Sorry, I'll repeat the question. Do we understand how the myosins align? And I think the myosin alignment is a secondary effect of tense cable are stretched, they are taut. And because they are taut, they recruit the myosin. So whether that's direct myosin sensing detention with a catch-point mechanism, or if it's indirect with, I don't know, other molecules playing a role of intermediate and recruiting the myosin, we don't know.
And you said that there was some useful and useless formins, kind of. Could it be that the formins that are not moving, like it's because they are pushing the actin instead of being driven by the actin?
So it's a fair question. When you look at actin or life act at single molecule levels, you never see them moving directionally. So maybe it is, but I wouldn't bet on it.
Thanks.
Okay, let's thank our speaker again. So our next speaker is Dr. Jack Bosma. He obtained his bachelor degree in math and physics at the University of Queensland, Australia. And later on in his undergrad, he found out his interest in biology, and then he pursued a PhD in biophysics at University of California, Berkeley. And he then stayed in the same institute as a postdoc to study new lifestyle imaging methods for visualizing transcription and production in real time. And he's now the PI in Health Institute in Netherlands, with a focus on investigating how non-coding region of the genome control gene expression in development by real-time transcription imaging. And let's welcome the speaker.
Thank you. Can you guys hear me? All right, I guess if I shove it into my beard, it's clear. All right. So, well, welcome. Good afternoon, and I'm really thrilled to be here. It's been a lot of fun getting to talk science to people again, and also to participate in some of the demos. And part of the reason why I accepted the invitation to come here is that this sort of had the vibe of a summer school I often went to in the States. Called Woods Hole, which is held in Cape Cod in Massachusetts, which is kind of this magical science place I encourage you all to go to. Sorry, I've got a bit of a scratchy throat. But there is one key difference, I think, between Woods Hole and here that I've found. The food here is significantly better. So despite the fact that the food at Woods Hole is not that great, I strongly encourage you all to kind of go check it out. So as you said, yes, my name is Jacques Bosma. I'm a group leader at the Hubrecht Institute, which is in Utrecht, about 20 minutes outside of Amsterdam. And I started my group there about 2 years ago, just before COVID, which was an interesting time to start. But we've done some fun things. But today I decided to focus on some published work, so to get into a little bit more of the detail of it. And then at the end, I'll tell you guys a little bit about some of the things we're currently working on. And and I'm going to be around until Thursday. And I've got my student Emilia Kadar who's here. So if you have any questions, just come find us and come talk to us. So the question that sort of drives me is I want to understand, how do the cells interpret the information in your DNA to make decisions? So all of us in this room started our life off as a single cell that underwent a bunch of different divisions, and now we're all about 10 to the 14 cells. And if you look at the nucleus of all of our cells, they've got basically the same DNA, right? So all the DNA is the same in all these different cells. But if you look carefully at the cells themselves, right, they're all quite different. So if you look at the cells in your brain, they look quite different, have very different physical properties to the cells that are coursing through your veins, and also the cells on your skin, right? And they need to in order to keep you alive. The cells in your brains need to conduct electricity, the ones in your blood need to carry oxygen, and the ones on your skin needs to keep stuff from the outside from getting in. And we've known that this is because they express different types of proteins, right? That's why they have these different physical properties. And this is to a large extent regulated by which genes are being transcribed, okay? By these other types of proteins called transcription factors. And so the classic view has been that there's a combinatorial code in terms of how you go and you define cell decisions and cell identity. So, a brain cell might express transcription factor A, B, and C, but not D, while as a red blood cell, you got a lot of D, but just a bit of A, right? So that's sort of the classical view. But more recently, people have started to appreciate that it's not just about which transcription factors a cell has, which determines its fate or its behavior, but that the actual dynamic information about how the concentration is changing in time can actually encode information for how that cell decides its fate or actually behaves, okay? And here's a couple of my favorite examples of where this is from a recent review, right? And this is one across, there's many different examples, but I'm just going to focus in on this one example, this transcription factor called P53, right? It's probably one of the most famous proteins on Earth. It's called the guardian of the genome. It's mutated in almost half of human cancers, right? And what happens is when you get some DNA damage, if P53 undergoes these oscillatory behaviors, right? Okay, the cell's in this mode where it's like, okay, I'm going to try and fix my damaged DNA, right? But if it undergoes this sustained expression, the cell dies, okay? So, and there's this beautiful little movie showing kind of these dynamics. So here, this dynamics is determining whether the cell lives or whether it dies, right? And then there's many different scenarios in between that kind of, where dynamics is really instructive for cell behavior. Now, people have done a lot of stuff for this in cell culture, and cells in culture are great. We can learn a lot of things about them, but for me, ultimately, I really want to understand what's going on in vivo. And to be able to do that, you really need to go and actually visualize these processes as they're happening in the animal, okay? And the challenge up until now has been that the same tools that we can use readily in cell culture is just not available in vivo. If you want to look at an actual critter that's developing, right, you kind of have to redevelop a lot of these tools. And so a lot of the things I did in my postdoc and a little bit in my PhD is to kind of try and develop these in what I consider to be a really helpful model system to understand sort of larger questions about development. And so let me introduce you to my model. So we work in the early fly embryo, okay? And flies are great for many different reasons. They've got great genetics, they're fast, I don't feel bad about killing them. So you can get a lot of them really quickly. So the fly embryo is great for many reasons. They're also very easy to image. You can shine as much light on them as you like. They're totally fine. And so the early fly embryo specifically is super great. And so what I'm going to show you here is the early stages of development. Okay, so this is sort of like a histone labeled tag that just shows you what the nuclei are doing. So to start with, what happens is you get these like coordinated synchronous divisions, right? Very, very rapid. These early divisions are all about just making nuclei, okay? And then you enter this final synchronous division where these cells now in this magical time called cell cycle 14, where they have 1 hour where they start off sort of pluripotent and then boom, they undergo morphogenesis, okay? So within this hour period, they have to go from being able to do just about anything to making cell fate decisions and then actually undergoing morphogenesis, right? So if we're going to try and understand how, this whole process works, we have to start at the beginning of transcription, okay? To be able to visualize how transcription is occurring in real time as this process is occurring, okay? And so fortunately, when I was starting my postdoc, a technique had recently been developed, which I think some of you guys have been working on in the modules. So for those of you who did, please bear with me as I explain it for the rest of the audience. To be able to visualize transcription in living embryos. Okay, so this was a technique that was developed in parallel in the labs of Thomas Greger, who was at Princeton, but is now split between Princeton and Pasteur, and Nathalie Destantny in Paris as well. And so the way it works is sort of like this. So let's say you have your favorite gene of interest. And what you want to do is you want to visualize the transcription as it's actually happening. What you do is you start off with the gene, and then you insert a stem loop sequence into the transcript. Okay, so this is a sequence that's pulled from a bacteriophage that when it gets transcribed into RNA, folds up into a very specific three-dimensional shape, okay? And this can get recognized by this other protein, okay? In this case, it's called MCP, this is MS2, that can then bind it with very high affinity, okay? And then if you create a fusion between the MCP and a fluorescent protein like green fluorescent protein, what you've essentially done by having this combination of this tag gene and this fluorescent MCP, you can now label transcription as it's actually happening, okay? And so if you go then and look at it in an embryo, so here's sort of an embryo with a bunch of nuclei here. When you look for a gene that's being expressed in the stripe pattern of expression, you look at these individual nuclei, what you'll see is a dot, okay? And that dot corresponds to the nascent site of transcription where you've got all these polymerase molecules bound to the DNA, so they're all locally enriched, okay? And how bright that dot is directly proportional to the number of polymerase molecules that are actually actively transcribing. Okay, so generally with the way that we do our imaging, we're not looking at individual RNA molecules diffusing around. You can do that if you like, but for us, transcription is much more of interest. And so I'm going to show you an actual still from a movie, and then show the movie. So where we've labeled the DNA in red. Maybe dudes with the lights, could you switch off the bottom one, if possible? Maybe not. So what you can do is you can see the DNA, the histones that have been labeled in red, and then the RNA is here in green, and you can see these individual foci of transcription. And then when I play the movie, what you're going to see, boom, cell division, transcription comes back. You can see lots of these fluctuations in terms of the actual intensity of the dots, and also the pattern, okay? So you can visualize these no, that was the other way. There we go. Thank you. I think maybe just keep it like that. I think my movies are prettier than I am. So it's less of a compliment for them than for me. We can now visualize transcription in real time in the context of development. Okay, and so that was sort of the state of affairs when I started my postdoc. But what I wanted to do is really be able to kind of go and connect all the way from transcription to sulfate decision to morphogenesis. And in order to do that, I chose to focus on this particular system, which Francois actually touched on briefly, which is gastrulation in the early fly embryo. Okay, so this is showing a snapshot of a fly embryo that's mid-gastrulation, which is the first, and many would argue, the most important morphogenic movement this fly will undergo in its life. Okay, so what happens is that it starts off with this, these cells are all on the outside, okay? And then at this point during gastrulation, the ventral most, the ones that will become the belly, okay, about 1,000 cells is a square, they all kind of dive in together, okay? And it's a very rapid process, it takes about 10 minutes, okay? And it's a highly coordinated process. They all have to kind of go in at roughly the same time, okay? And this process is orchestrated by this transcription factor called snail, okay? And the surprising thing is, from the onset of snail transcription to the induction of this coordinated morphogenic process is about an hour, okay? It's really, really fast, okay? And the nice thing also is that snail is deeply conserved across the metazoa, right? So you have snail, and in fact, if somebody develops breast cancer, one of the markers that is checked for the ability to metastasize is snail, because snail also drives the epithelial to mesenchyme transition in many other contexts. Okay, so wanted to understand, okay, but can we understand how this process happens so rapidly and so coordinatedly? Okay, so snail induces the coordinated transformation of movement of these cells inside. The other thing that snail does is it creates a very precise sulfate decision. So what happens when you look at snail to start with, sort of the middle of the cell cycle 14 period, it's present in this gradient of expression, where there's high levels in the ventral part of this embryo and then progressively lower levels towards the dorsal part. Half hour later, though, what happens is this pattern sharply refines, okay? Where you have cells that are expressing high levels of snail right next to cells that are expressing none, okay? And so these cells are the ones that are going to dive into the inside of the embryo, and right next to them, those cells are going to turn into neurons, okay? So this sulfate decision has to be razor sharp. It also has to do with this epithelial to mesenchyme transition, where the cells, they either have to commit or they have to not, okay? So snail has to be kind of like expressed in this highly coordinated pattern, okay? And it also needs to make this really sharp boundary of expression. Okay, and so how does that work? So, we know that it's a dynamic process. Can we visualize transcription, and can we learn something about this process from that? So we used this new technology to be able to visualize transcription, and we went and we tagged the endogenous snail locus, okay? And similarly to what I was showing you before with that other gene, what we were sort of surprised at, so we thought maybe snail was somehow a little bit different and special, is that when we looked at the foci of transcription for snail, they were just as messy and erratic as all the other genes we'd looked at. So if you look at this border here, these cells are sometimes they're switching on, they're switching off, their intensity is all over the place, okay? So you have this gene that's involved in this really fast, really tightly coordinated process whose expression is sort of a mess. Okay, so and you could go through and carefully quantify the levels of expression in individual nuclei, and you see these bursts that are quite typical of how genes are transcribed. Okay, but so you have these enormous fluctuations. Okay, and the thing is, this gene is very unstable, both the RNA and the protein. So it's not a matter of the time just averaging everything out. Okay, so we were quite perplexed by how can you have this gene that's being transcribed in this really messy fashion, okay, within an hour from its onset actually coordinating this highly synchronous process and well-defined sulfate decision. So, had to remind ourselves, much as I love transcription, okay, it is an important but it's only the first step in driving cell fate behavior, right? We just saw before, so the actomycins are the ones that are actually doing stuff, right? So we had this tool to be able to visualize transcription, and that's great, but then that has to go into RNA, which then has to go into protein, and then Snell has to regulate other stuff that regulates the actomycins. But Snell's key role is here as a protein to switch things on and off, right? So ultimately what we want to be able to do is actually see the Snell protein, okay, in real time. So this is a sort of a fixed embryo showing the Snell protein here, right? But there's a problem, okay? The challenge is that fluorescent proteins mature too slowly to visualize transcription factors in real time. And let me walk you through why that is, right? So, if let's say you want to tag your favorite protein of interest, right? With CRISPR nowadays, you just knock in something like GFP, right? I mean, it's easy these days, right? The problem is that from the time it takes from when your protein is made to when it starts to glow, that you can actually detect it. For GFP in the fly embryo is about 40 minutes, okay? It's a while, but I'll go through some other ones in a second, and they're similar, okay? So there's a substantial delay between when the protein is made and when you can actually see it. This problem is further compounded by the fact that this protein, Snail in particular, many other proteins in the fly embryo, is very unstable, okay? It's actively being degraded, right? It has a half-life of about 10 minutes, okay? So it means that for most of these protein molecules, they're dead long before they've ever had a chance to glow, okay? So you're in this very challenging situation. There's these delays, and then you can't see them because they're degraded very quickly, which makes it very difficult to tag fluorescent proteins and actually use them, okay? So one might think, okay, well, this seems unusually small, but whenever you've got a dynamic system, okay, the time scale in which you can respond, the fastest you can respond, is set by this, the degradation time of your protein, right? So there are many other systems where, proteins are actively degraded. It might seem energetically stupid to do that, but what you get for that is the ability to move quickly. So just to kind of show you that this, it's 40 minutes in the fly embryo, we carefully quantified this. But this is not unusual in terms of how long it takes fluorescent proteins to mature, right? So this is really cool. If you ever had a chance, you should look at this paper. They did some really cool work where they quantified the maturation time of a whole bunch of different fluorescent proteins in E. coli, which is not quite, flies or worms or whatever, but it's pretty good. And so what you see there, so they do it at different temperatures. Obviously, it's temperature dependent, but monomeric EGFP, it takes, 32, 60 minutes for 90% of it to mature, right? If you go down to the YFPs, there's some YFPs that are better, monomeric Venus envy, you've got, it's 20 minutes, okay? But there's this trade-off. The yellows, if anybody's worked with the yellows, they tend to be a lot less stable, okay? And a lot less bright. And, I think Nathan's, he's gonna shake up this table quite a lot, clearly from the talk he gave. Okay, but I think fundamentally we're never going to get to the point where we're going to be able to make a fluorescent protein that can mature on the order of minutes and be really bright, okay? And the reason why that is because there's basically a bit of a catch-22 when it comes to whether you're bright or whether you're rapidly maturing, right? So here's this beautiful structure of GFP. It's just, this epitome of structure-function relationship in proteins where you've got this beta barrel that's cradling this chromophore, okay, and it stabilizes this chromophore in such a way. If you take this chromophore and you just put it in water, you don't see anything, right? The protein is what makes it fluoresce, what allows it to fluoresce. But now, to go from a protein, once this is coated and it folds up into beta barrel, which happens really quickly, to something that glows, you need to have these chemical steps, right? Cyclization, dehydration, and then oxidation is the slowest part of this process, okay? And so the catch-22 comes from this. Okay, so to be bright, a protein has to shield the chromophore from solvent and oxygen. Oxygen is bad if you're a chromophore, right? But to mature rapidly, the chromophore needs ready access to the solvent and oxygen so that the oxygen can get in there and actually catalyze the change, right? So unfortunately, we're never going to be able to make these fluorescent proteins that can do both. So there's always a bit of a trade-off, okay? And so if you're looking at highly dynamic processes, this creates a problem where, so this is kind of a simulation showing you in green the fluorescence you get from a protein with a maturation half-life of 40 minutes and a half-life of 10 minutes versus the actual total protein, okay? So the first thing that you notice is that it's way lower, right? So because protein maturation is it's a stochastic process, it's exponentially distributed, no matter what the half-life, you'll always have some of them that mature fast enough for you to be able to see, okay? But as these numbers get larger and larger apart, or even when they're similar, the number of them that happens decreases significantly. So your signal just gets really dim, okay? And this is typically what people see. Often, when you have a highly dynamic process, you tag it with a fluorescent protein, it'll just be really dim, And the fly, often when people tried this, it just wouldn't see anything, okay? The other thing is that it's a little hard to see from here, but these two curves don't scale, okay? To be able to go from protein to fluorescence, there's this highly nonlinear, time-dependent correction, which depends very precisely on the production term, okay? So in order to try and visualize protein dynamics, we had to develop a new tool to be able to do that. And that's sort of what I did in my postdoc. And I'm going to tell you what we did to get around this challenge. And so let me set up the technique for how it works, and then we'll go over what we were able to do. So to start with, what you do, we use this llama tag. So the reason for the name will become obvious in a second. But what you do is you start off with this situation where you use a maternal driver. So the mother puts in mature green fluorescent protein into the egg. It's uniformly distributed at low concentrations. Okay, so if you look at an egg, it's just sort of faintly green uniformly. Okay? And then what we did is we took these single domain antibodies from llama, just a 100 amino acid piece at the end of these antibodies. So there's a quirk in llama immunity, which I'll go into more. But basically, from a llama antibody, you can take the end 100 amino acid piece. It will maintain its binding affinity, which is very high, in this case to GFP. You're talking nanomolar, very high on rates, okay? And it actually, when it binds GFP, actually increases the fluorescence, okay? So if you take that and then you make a fusion between that and your transcription factor of interest, okay, use CRISPR or whatever, which we did in this case. Hunchback is the transcription factor I'm going to talk about in a sec. It doesn't matter the details. But what happens is whenever this protein is translated, okay, this antibody, the llama tag, okay, it will grab onto already mature GFP, increase its fluorescence, and because it's a transcription factor, we'll drag it into the nucleus, okay? And so the fluorescence you observe there will then increase quite significantly. And so when we implemented this, we use a CRISPR fusion to tag endogenous hunchback. It's this transcription factor that's expressed in the head, the anterior region, a little bit in the tail. We could see nice signal, okay? So we were very happy that we had good contrast. We could actually see that it was working. And we were then able to actually visualize for the first time Hunchback's dynamics. Hunchback is more of a control. It's not super duper dynamic, but it was a great control because a lot of people have studied it. So here, we have this maternal GFP and this Hunchback llama tag, and you can visualize the dynamics on the minute kind of time scale, right? And so this was great. We could quantify the protein concentration, what we thought was the protein concentration in real time from the fluorescence. So yeah, so you can see the fluorescence. The pattern looks good. But does the fluorescence that we're actually measuring reflect well on the actual protein concentration? And to be able to test that, what we did is we took these embryos that I showed you before, okay, and then we chemically fixed them. And then we counterstained using an antibody that just recognizes the hunchback protein, which was stained in a different color, okay? And then we went and quantified the intensity in each of these nuclei. So this is with the authentic, with a protein stained against the protein, an antibody stained against the protein, and this was just a green fluorescent signal that we were seeing in the nuclei. And we were very happy to see a nice linear fit to that, okay? So what we were seeing with our fluorescence was directly proportional to the amount of protein by immunofluorescence. There's a little offset here in green that has to do with the free GFP concentration that's floating around. So we know it works well, and you know what I showed you earlier was in the early fly embryo, but hunchback, luckily, it is more interesting actually later in development, where it's an important transcription factor that's involved in specifying neural fate identity. So the fly nerve cord does this cool thing where you'll have neuroblasts that express a successive series of transcription factors, and depending on which transcription factor it expresses will determine the fate of the neuron that it gives rise to. So what happens is these neuroblasts start by expressing high levels of hunchbacks, then it goes to low levels of hunchback, and then it goes to no hunchback, and then something else takes over. And so what we were able to see is we could see those dynamics in our movies. So here is just a zoom in of a fly embryo. We're looking at the surface. We're going a little deeper in each stack, and these big cells are going to be the neuroblasts, and you can watch how they're increasing their hunchback levels of concentration, and then decreasing it, and then eventually it goes to nothing as these guys go on, and you can see the ganglion mother cells also express it. So basically, I mean, the key thing here is that it works later in development as well. And so we were super happy to see that. And this was actually, I mean, this was one of the in the paper. We didn't have this in the original version of the paper, and then we had a really cool reviewer who was like, "You should do that experiment." And then we did the experiment, and it was really helpful. So sometimes reviewers can be very helpful. And so so we showed that. Now the other thing that's neat about the platform is that it's very easy to multiplex. So what we were able to do is we were able to just take some other antibodies from llamas that people have made against M. cherry, okay, and then just use that instead of an antibody that was made against EGFP and tag 2 proteins at once, okay? And so you can multiplex it very, very easily, and you can follow these 2 proteins in real time. Okay, so that's sort of the technology, and since I thought this was a school, we'll go dig into a little bit more of the detail with, because I know there's a lot of other tagging technologies around there that some people might be thinking about, so I thought I'd go through and walk through, how this one is different to other ones that might be relevant to people. So in terms of the different tagging technologies that exist out there, the main difference as far as I see it is protein stability. Okay, and that might be a little, you're like, why protein stability? But let me walk you through it. So most of these approaches that people use to tag things in vivo, right, rely on antibodies, right? It's this magical thing, you inject an antigen into a critter, and you get this protein that recognizes that antigen with very high affinity. Okay, and it works on all creating these, these antibodies as a form of a human, but a mouse one will look basically the same, where there's like lots of different variants of these proteins at the very end. This is where the antigen binds, right? They do all this recombination shuffling to be able to find one that works, right? But now the thing is, so this kind of, here you have, in humans, you have this heavy chain, which is over here, and then you have a light chain here, and you have this link, okay? And that link is actually this thing called a disulfide bond. So you've probably heard of these things called disulfide bonds. They're an important thing where 2 cysteines bind together, right? So there's a disulfide bond there to be able to link the heavy and the light chain. But when you actually look at disulfide bonds all over the thing, these things are riddled with them, okay? So all in all, you have about 12 disulfide bonds. So you have about one here at the start, one here at the beginning. So there's disulfide bonds to kind of link everything together, but they also stabilize the whole structure, okay? So they're really, really important, okay? But the kicker is that these don't exist in the cytoplasm or the nucleus, okay? So the inside of the cell is a reducing environment, okay? And so this is why when you try and express one of these antibodies or even put it inside the inside of a cell, it just falls apart, okay? Because it's missing all of these different disulfide bonds, okay? And so what people have done to try and create, you call this, there's these variable fragments that people have created to try and capture the antigen binding affinities of these things, right? So you've got the minimal piece to be able to bind. What happens is when you express that intracellularly, just for comparison, you're down 3 disulfide bonds, okay? And so you can go and for, to first order, roughly speaking, what that means in terms of your stability is that you're down about 30 degrees Celsius for your melting temperature. Okay, so that's a huge destabilization of the protein, right? And this is why for when people need to try and make these, they need to do a lot of protein engineering to try and build in, stabilizing forces that are sort of equivalent to what they had with these disulfide bonds. And that works to varying degrees, and often people have to add other proteins that are expressed in the same peptide to stabilize it further. And so, and it kind of works, but these things are still inherently quite unstable. And so this is where, so the Suntag, some of you might have heard of, the Frankenbody, there's a whole bunch of different techniques that kind of rely on this kind of approach. And so how does this compare to kind of llama antibodies? So llamas have this weird quirk in their immune system where they don't have a heavy and a light chain, okay? They just have a heavy chain, okay? So all of the antigen specificity comes from this one piece right at the end there. Okay, and so if we now look at, are there disulfide bonds? Yes, there are disulfide bonds here too. Okay, you can see them here. There's, instead of having 12, you only have 8 because those 2 are there. But you have fewer of them, okay? But then what you do, you can snap off the end of that here like we did when we were talking about using the nanobodies, okay? So the thing is, these also have a disulfide bond missing, okay? But they only have 1, okay? So because they only have 1 disulfide bond missing, their drop in stability is about 10 degrees Celsius, okay? So that means that normally with these single variable fragments, you try and express them in the cell, they don't do anything, they just aggregate, okay? You gotta do some protein engineering to try and stabilize them a little bit. With these guys, generally, once you put them inside the cell, they work pretty well, right? Again, there's been a hit in the stability because you're missing a disulfide bond. And with some of the nanobodies, that's not enough, right? And it's also quite temperature dependent. So one of the nice things about flies is they exist at room temperature, right? So their proteins, if, when a protein in mammals might be unstable, it can be totally fine for flies, right? And so this is sort of the, at least as far as I see it, the big difference is sort of the stability with these things, right? And so one of the big things for us when we were developing the technology is, we wanted to work on transcription factors that are not the most stable proteins out there. And we wanted to be able to make sure that they were functional, okay? So it was really important for us that the thing that we were using to tag them with was as stable as it could possibly be. And so there is this intrinsic stability, but then there is also an extrinsic stability that has to do with how you set up your experiment. Okay, so here I'm showing you a crystal structure of the llama tag, which is this nanobody that binds to GFP, bound to GFP over here, right? So again, it's beautiful GFP structure, and this is the nanobody here. And what you can see, and if I had multiple hands, I would show you, is sort of like there's a large surface area of interaction between the nanobody itself and GFP. It's sort of like a hand that kind of grabs hold of GFP, okay? And so there's a lot of surface contact between it, right? And so the way we also designed our experiment, we have it such that this nanobody is associated with our protein, right? And we have this free GFP. So, oh yeah, sorry, I forgot to say. So this large surface area contact, okay, this stabilizes this even further. Okay, so this is a large form of stabilization actually. So when this binds to the GFP, the complex is a lot more stable than the isolated protein. Okay, and so by design for the way that we do things, we have this nanobody that we then fuse to our transcription factor of interest, okay, and then we have this free floating GFP, which means that as soon as this thing is produced, there's a GFP ready to bind to it. Okay, so it means that it might be unstable a little bit, okay, but GFP binds and stabilizes it very, very quickly, okay? But if you look at, for example, this is true of many of the variable fragment techniques, but the Suntag here is just chosen because I got this crystal structure. Okay, here's sort of the variable fragment, the crystal structure of the variable fragment. There's these two pieces, like I was saying before, and then you have this alpha helix, which is the Suntag, which is the GCN4 region. Okay, you can see here that the surface area of the interaction is much smaller. Okay, so this binding interaction doesn't stabilize things nearly as much as this one over here. Okay, and then a lot of these approaches, what ends up happening is that a very small fraction of the binder is actually bound to the ligand. Okay, so there's not a lot of stabilization that happens, and this is why often when these single variable fragments are expressed intracellular, they need to have a lot of stabilizing proteins bound to them, okay, in order to keep them from aggregating or. But the other challenge is that even in this context, for unstable proteins, they can be quite problematic, okay? So it might seem like I'm trashing the Suntag approach, which I'm definitely not. I guess the thing is here, you have to think about what's really important for what you want to look at, right? So the Suntag approach has been used by, my colleague Marvin and also by Munya Lagat, who some people in the audience are from her lab, to visualize translation really, really well, okay? And if you're really interested in translation, right, you want to see that, that little peptide as it's just popping out of the ribosome channel, right? This is the way you got to go, right? Because this can find that little, alpha helix as soon as it's made and bind to it, right? Like with the LlamaTag approach, right? If you try to do it with like either GFP or the LlamaTag, the whole protein has to be synthesized. It's, it's slower, okay? But the trade-off is stability. Right? So you have to really think about, okay, well, what's important for what I'm trying to do to understand what some of these techniques are appropriate for you. Okay, so that's sort of the methods part. And so we're going to jump back to Snell. So to remind people, so, it was this question, this transcription factor makes this boundary, drives this invagination process. We're trying to understand how we had this stochastic pattern of transcription turning into a reliable, reproducible pattern of protein and then morphogenesis. So what we did is we used the LlamaTag to tag Snell. The first thing that we were very excited about was the fact that we could see it. So people had tried to make GFP fusions with Snell before and they just wouldn't see anything because of this maturation issue. So we saw that, okay? And then when we looked at its dynamics, we actually learned a bunch. So what you can see here is you don't have the same degree of fluctuations that you do with transcription. And then when you look at the boundary here, you're seeing these cells are very rapidly making their sulfate decision, They go from high to low very, very quickly. And then boom, you get this morphogenic movement. So, you're looking at the protein that is upstream of everything that drives that morphogenic process, okay? And so what we wanted to do was really nail this down by being able to look at both transcription and the actual production of the protein itself. Okay, so we combined this MS2 system, which allows us to visualize transcription. Okay, we stuck the MS2 tags in an intron. Okay, and this LlamaTag system that allows us to visualize protein. So what you're going to see here is you're going to visualize transcription, okay, in red. Okay, and then those RNA molecules are going to make protein, which you can see in green, and then you're going to see gastrulation, which is the morphogenic movement that is driving. So you're going from all the way from the locus to the cells moving, okay? And so you can see here the RNA pattern is transcribing, super stochastic, okay? But the protein pattern is not nearly as stochastic, but you can see it's sort of in the beginning, and then they sort of coincide, and then boom, you get morphogenic movement, okay? So it always just blows my mind that we can now go from, watching what's going on at the level of DNA to seeing this, the actual cells. So I'll slow it down a little bit. So one of the things that jumped out at us to start with was that the protein pattern was further away than the RNA, and then they started to coincide. So that was a little weird to us. And we're like, okay, well, what's going on there? So this is so if you look at a still, you can quantify this more carefully. So your protein is further away than the RNA. And because we can see the RNA, we can visualize transcription all the time, we know that there was never any transcripts there, okay? But then, half hour later, the two coincide super duper well, right? So what's going on? And so one of the things that, which should have been obvious at the time, but so what happens is that the early fly embryo is what's called a syncytium, okay? So you have these nuclei, okay, that are all in sort of a shared cytoplasm, and what happens is that during cell cycle 14, this 1 hour window, membranes are actually coming down and taking this and turning each of these individual nuclei into a cell, okay? So during this process, during this time, they're all sort of potentially dynamically coupled. They can share things by diffusion. Okay, and I'm going to show you this movie of these myosins, which are just at the ends of these channels, and you can see in dark here is the nuclei, and you can watch as the myosins drag the membrane past these nuclei and form cellularization. Okay, and so pretty movie. And what we can do now is we can go through and actually, because we have this construct where we can visualize transcription and we can visualize protein, we can go through and carefully quantify this effect that, that I was mentioning earlier, where you have protein that extends beyond the RNA pattern. So we can go and we can choose a cell like the cell number 1 here, where it has transcription, and we can quantify the protein concentration in it. It increases in time. That makes sense. And similarly, we can pick a neighbor of it, okay, that never shows RNA, this nucleus 2, that never shows any transcription, okay, but it has protein, okay? And we can look at, okay, what is the dynamics with that, okay, and then we can, based on that, we can actually quantify how strong is the coupling between individual nuclei, right? And so when we do that, we were sort of very surprised to see that the coupling, to start with, is really strong. Okay, so there we get this number of 0.5. So what does 0.5 mean? It means that in a given nucleus, okay, half of the Snell protein in that nucleus came from transcripts produced in other nuclei. Okay, so there's an enormous amount of sharing through diffusion. We don't know whether it's the protein or the RNA. Okay, could be both. Of protein like that. So, the amount of transcription factor you have in a given nucleus strongly depends on what you're doing, but also on what all your immediate neighbors are doing. Okay? And this is also time dependent. So the coupling is initially quite strong before the membranes come down, and then as time goes on, it sort of decreases significantly. And so it turns out this coupling being time, that's not the only time-dependent thing. So this is sort of, a picture of the snail boundary here. And turns out snail's regulation is also time-dependent. So you start off with snail being driven by these ubiquitous strength factors, twist and dorsal in this gradient. And then as time goes on, so during this 1 hour window where this coupling strength changes in time, you get this kick-in of this autoregulatory function. Okay, where the snail protein is necessary for snail transcription. All right, and so when you pull these processes together, what it means is you can end up in the scenario like this nucleus where we have here, nucleus number 3, right, where what you'll have is a nucleus that doesn't show any transcription. Okay, not transcribing for whatever reason. Okay. But it has a whole bunch of nuclei that are transcribing and making protein. So when you look at its protein levels, it's increasing, even though it's not transcribing at all. Okay? And then eventually, okay, it sort of kicks on because it's got enough snail transcripts. Okay? And so the ability for that individual nucleus to start transcribing is heavily dependent on what its neighbors are doing. So there's a sharing of information, and it's through diffusion, which is basically as fast as you can go in these systems, right? And so the idea is that it's sort of the rapid cell decision here is defined by this coordination of the time-dependent coupling, okay, and this timing and the switch of regulation. So the idea is you start off, you've got messy transcription, which is leading to RNA, and so you've got this diffusion of either protein or RNA, or both, doesn't really matter, that kind of leads to this kind of graded pattern, okay? But eventually you need to make a discrete pattern because, snail has to catalyze these two fates, right? And so what happens is as time goes on, you get these membranes coming down, okay, which is decreasing the degree of coupling between each of the individual cells. And just as that happens, okay, and during this time, right, the amount of snail protein you have depends heavily on what your neighbors are doing, okay? So there's this sort of quorum effect thing going on. And then as time goes on, this autoregulation kicks in to lock in this kind of fate decision that's sort of based on what all your neighbors are doing, okay? So it's a way for you to average out stochastic fluctuations, okay, rapidly, but then also eventually lock in a discrete cell fate, okay? And so this is sort of how we've drawn this in 2 dimensions, okay, sorry, in 1D, but in 2D what you'll see is, so the idea is when this propagates out on this grid, the idea is that's how you kind of go from this edge of RNA transcription, which is sort of messy, okay? And RNA looks like transcription is just sort of noisy and bursty by nature. It's just there's nothing you can do about it. And then by having this kind of time-dependent system, you can go from, messy RNA, and 30 minutes later you can have a discrete, well-defined cell fate decision that drives coordinated morphogenesis. And so it's sort of one thing to understand about the fly is that, when you look at the fly, it's a system that's really optimized to go fast, And you're like, well, why does time matter so much? Why is half an hour really, really important for the fly, okay? So for flies, speed, really is a matter of life or death, and you're going to ask why. Okay, so flies, fruit flies, which you've all had in your kitchen, I'm sure, they're originally, like myself, from Africa. So I grew up in southern Africa, and they're actually from the woodlands of southern Africa, where their favorite fruit is marula. Okay, so it's this particular type of fruit, and people have done these test experiments. You can even take, wild flies and have an apple or an orange and then a marula, and the flies will always go for the marula. Like it's not even like 10%, it's like 99% of them do it. And even the lab strains, they haven't forgotten their African roots. They still go for the marula. So some of you might remember, I don't know, there's this alcoholic drink called amarula that it's sort of based on. So the thing is, so the flies, they have to find this fruit and then lay their eggs on it, right? And then the larvae have to come and develop on it. Now, it's a transient fruit source, right? So it's expiring away, so you got to develop quickly. There's competition from individual larvae for each other, right? Okay, so that drives you to be fast, okay? But the other thing is this, in these African woodlands, there's not that much fruit around, okay? And there's a lot of things that like to eat this fruit, okay? But in particular, elephants, okay? They love this fruit, okay? So if you're a fly embryo, right, sitting in a marula, right, you gotta go fast, otherwise you're gonna end up there, right? And then it's game over, right? So there's a lot of pressure on these critters to develop really, really quickly, and they've been under that selection pressure for a long time. So this is why we think it's what's sort of going on. And I realize I'm running a little over time, so just briefly, I'm going to talk about some of the things that my lab's sort of interested in working on, looking at transcription factor dynamics, looking at transient transcription factor dynamics in small spatial clusters and how that regulates genes. And then we're in the process of building new tools to visualize protein-protein interactions. And these are some acknowledgements for the work that was done in Berkeley. Particularly Hernan Garcia, who was a great mentor, who didn't believe the llamas were a great adjustment to flies, but he let me do it anyway. And this is my lab, funding, and at my research institute, we're hiring this year. So strong emphasis on imaging, wonderful place to start a lab, not that far from here. So if anybody wants more interest, or is interested, come talk to me. And I think I'm done then.
So we only have time for 2 questions at most. Okay.
So thanks for this amazing talk. So I'm just puzzled by the role of snail. So we know that snail transcription decreased during nuclear cycle 13. So it was published, I think, by you. That snail is a repressor of that snail protein is a repressor of snail transcription, and now in your models you are showing us that snail is an activator of transcription. So I'm a bit puzzled how you imagine this role.
Right. Yeah. So snail classically is an active, is a repressor, and it's true that snail levels, there's some dynamics there, but on average it's going down. But Maria Lepton has published some stuff showing that it seems to be like a lot of transcription factors. It can be an activator or a repressor depending on what its context is doing. So I don't know definitively whether snail is a direct activator of itself or whether it's snail is repressing a repressor of snail. But for us, doesn't if the action is indirect, that's all that matters. So the evidence that it is activating itself directly or indirectly is very clear. If you take a snail protein null, or where it's unable to bind the DNA, you initially have transcription, and then it goes away. So it's very clear that functional snail protein is necessary for maintaining its expression. But whether it's direct or indirect, we don't know definitively.
Okay, thanks.
Last chance. No question? Okay, let's thank the speaker.
Thank you very much.
Just a little reminder, at the beginning of the next session, just before the speech of Jörg Enderlein, we will have a short presentation for MIFOBIO YSN, and then it will be the poster awards. So please come.
And your microphone is there. Yes, and your microphone is there.
Yeah.
I put open media folder.
No, for now it's good, and I just know there's something. So, hello everyone. I think we will continue with the last seminars and our last presentation. Okay, so good evening. I would like to present you shortly MIFOBIO YSN, which is MIFOBIO Young Scientists Network. So it's a group which was established in 2018 by initiative of young scientists, young PhD postdocs. And actually our group, as you know it today, we continue this work. So basically, who is present here? It's me, Clément, Valerio, Nathan, and Samer. Most of us you already know. And so, why do we do that? What are our goals? We want to bring together young scientists around MIFOBIO, around bioimaging, which we mean PhD postdocs, but also young engineers. I mean, we are really open to we want to widen this network, and young PIs also. Well, okay, we let judge you what means young. Okay, this is up to you. But I mean, we are really open, and we really want to keep going this network and build solid base, which can also help, for example, organize this great event as MIFOBIO is. Okay. Then when you have some network, it's much easier to increase knowledge transfer and enhance scientific collaboration. It's difficult to do collaborations if you don't know anyone. So this also can help you, especially if you're at the beginning of your career. And in the end, yes, we want to also enhance interactions with industrial partners. As for example, we had the round tables also a little bit about it, and there were people who work in the industry. And also, because within the JDR MIFOBIO there's many labs, we want to encourage the interactions between these labs. And our group, we organize every year a conference, so this time it will be in summer 2022. We will start organizing just after MIFOBIO. And it's a really nice conference, and last year it was online, which was a bit sad, so we're really looking forward to make it in person next year. You will get more information about it soon. Okay, so during MIFOBIO, we think it's a great opportunity to recruit new people to our team, because I mean we are not many, and we would like to do maybe more activities, but we need more partners for this. So if you are interested, please write us an email and tell us, yes, I want to join you. So you can write an email. If you forget the email, you can find it again on the website of MIFOBIO. And if you have questions, we thought it could be a good idea to, if you want, you can meet with us for lunch tomorrow, which means Wednesday, and we will be at the back of the restaurant on one of the tables, and you can join us and ask us any questions you have, and we will be happy to discuss with you. Okay, and I think we can move to the poster awards that you are all waiting for. And for this, I will pass the word to my colleague Valerio.
Okay, so now is the moment to give the prize to the best poster. I want to thank Anna for the really nice introduction. I just want to add one little thing. You can join ESN, our young network, even only to remain in contact with us, form this kind of network of collaboration and talking. It doesn't mean, if you join, doesn't mean that you have absolutely to be an organizer. So we will force you at one point, but that's another game, let's say. So someone told me that it's really lonely to give prize alone, so I would like to call Marie Fourier on the stage to help me with handing down the prize. So Okay, thank you for arriving from the yeah. She probably was leaving Balambra while we call her, I don't know. But okay, so we can start. As usual, we start from the third prize and go up to make things more complicated and fancy. So for the third one, I would like to call Laurent Lobj. I cannot say the surname, sorry. Okay. So the prize is yeah, you will. You can give it. Will not bite. So, I will invite you to introduce yourself 1 minute briefly.
In French.
Whatever you like.
Ok. Bonjour à tous. Alors, moi, c'est Laurent Nobjoie et je suis ingénieure d'études sur le plateau d'imagerie à Infinity à Toulouse. Et je travaille sur la transparatisation d'organes. Donc, voilà. Je fais les acquisitions sur du brucaire de photons. Et du coup, j'ai fait les acquisitions et mes expériences avec ma collègue, Lucie de Mersman, qui n'est pas là, mais que je remercie. Et aussi Sophie Allard, qui nous a bien aidés sur les projets, mais aussi sur la construction du poster, qui n'était pas si simple. Et donc, sans elle, ça n'aurait pas été possible. Donc, merci autant à Sophie qu'à moi. Donc, merci à tous pour les votes.
So, thank you a lot. Maybe you can remain here so we can take a photo of the winner at the end. So, second one is Marco Paoli. Oh, you can. Important role, you see, it's really like are we sure we want to say the prize? What is it? It's not a secret?
You don't have to say it completely, but the third prize are goodies from Stem Cell Company. The second prize, it's a gift voucher for Decathlon.
Don't say the first one, wait.
Yes, okay.
We'll tell you later. So, again, if you want to...
Okay, well, I'm Marco. I'm a postdoc at the CRCA in Toulouse. It's a Center for Animal Cognition Research. And it's the first time in MIFOBIO, so I was quite glad to see all this interest for my topic. And well, thanks to the organizer and to whoever voted my poster. C'est ça? Merci.
So now we go for the really big first prize. I don't know actually if you want to give it out or we can keep it. Forgot now. Okay. And it's too late to put my name now. So if you want to say what is it, it's a free stay.
And the third prize, it's a gift voucher for holidays at Belambra, which we can select as you wish.
Well, bonsoir à tous. Je vous remercie. Tout d'abord, je vous remercie. Je suis Ali Ahmad. Ou bien vous pouvez m'appeler Ali Ahmadip, comme mon superviseur David Rousseau aime m'appeler. Je suis un ingénieur de recherche à l'INRAE à Angers. Je viens de finir ma thèse en septembre. J'étais au laboratoire Creatis. Nous travaillons sur le développement de simulateurs des images microscopiques pour les cellules uniques et le développement de méthodologies basées sur le traitement des images, surtout en deep learning pour le traitement des images des cellules uniques. Je vous remercie tous et si vous avez besoin de plus d'informations, nous pourrions discuter après à côté de mon poster qui est le numéro 67. Merci.
Okay, and to say thank you to Marie, we actually have a little prize for you. It's not fancy like the one from Abelite, but you can use it at home, I don't know, to do stuff. So enjoy it. And basically, this is the end, I suppose. Yes. So if everyone wants to come in the middle, you can take a photo all together with the prize.
Can you take a photo?
Like,
I just want to add that Marie Fournier, she was really at the first group, and we really want to thank her to have the initiative to establish MIFOBIO YSN. And the first team, they really did a great job, and we really want to continue.
Okay, so now we can empty the stage and give the microphone to the next speakers. The chairman is... okay.
Okay, so now we have the pleasure to welcome Jürgen Allein. So he did his PhD in Berlin, and then he did a postdoc at Los Alamos before going back to Germany and taking a position of assistant professor. And he's now professor for biophysics at the University of Göttingen. So probably you know him already for his work about single molecule spectroscopy and super resolution microscopy. And two of his main topics of interest are lifetime measurements and interactions between surfaces and fluorescence or fluorescent molecules. And today his talk will deal with both, since he will tell us about how to extract molecular actual information from lifetime modification induced by the proximity with metal or graphene surfaces.
No? Yeah. Okay. Yeah, thank you very much for this very nice introduction. And I have to thank the organizers to invite me here to MIFOBIO, which is my first MIFOBIO, and I had no idea how great this meeting is. So my French friends, they all urged me for many years to come here, and this time I made it, and it's fantastic to be here. Thank you so much for giving me the opportunity to present my work here. Now, of course, because it's a microscopy meeting, my talk will be about imaging microscopy. So, as you have seen, I guess this is now common knowledge, we have a resolution limit in classical microscopy, which is the Rabi resolution limit, telling us that the lateral resolution that we can achieve with a normal good wide field microscope is the wavelength of light divided by 2 times the refractive index, and sin is the half angle of the light cone of detection of the objective. And here this is only an animation showing you how the coherent emission of 2 emitters looks like when they are coming closer and closer. And at some point your objective simply loses the ability to distinguish between these 2 emitters, and this is exactly the Rabi resolution limit. Which is not so well known is that we also have a resolution limit, the corresponding one along the optical axis. This is the axial resolution, which you basically see when you have a confocal microscope, and it gives you again a number telling you how good you can distinguish 2 emitters along the optical axis. And this is given here as the wavelength of light divided by the refractive index, and now 1 minus cosine this theta angle, the half angle of the light cone of detection of your objective. And the interesting thing is when you go to, this is again here simply a visualization how it looks like, and what you can see is when we compare the lateral and axial resolution, and here also take into account the different wavelengths, for example here starting from 700 nanometer to 500 nanometers, then you have this band here for the axial resolution and here for lateral resolution as a function of the numerical aperture of your objective. What you can see that there's a big difference between the axial resolution and the lateral resolution. Of course, this is familiar knowledge. When you have any experience with confocal microscopy, you are familiar with these anisotropic image resolution in your images. And even when you go to very high resolution, you still have a factor of something like a 3 to 5 difference between these two resolutions. An interesting point is that when you go to super resolution microscopy, and for example 3D stat or these fantastic methods for single molecule 3D localization like astigmatic imaging, biplane or PSF engineering, you still keep this ratio of factor 3 to 5 between the external and resolution, so that in typical microscope settings and with typical photon budgets, you get an X resolution of 30 to 50 nanometer. The only exception to the rule are the interferometric methods like interferometric palm or isostat, which uses interferometer for looking on the sample from both sides, or exciting from both sides. They can indeed achieve then an isotropic resolution in the order of 1 to 5 nanometers, or then the recently 3D Minflux also is claiming to get the same isotropic resolution. However, as you may know, these methods are nice, but they are extremely technologically complex. So there is one way we can do it much easier and simpler under specific conditions when we use near-field optics. And one method I want first to present here, this is variable angle total internal reflection, fluorescence microscopy. So I guess you are mostly familiar with TIRF microscopy, where you excite your sample in a wide field setup with a plane wave on a very high incidence angle, and the bigger the incidence angle is, shown here, then you get a more and more steeper decaying excitation intensity above your cover slide. This is the TIRF microscopy idea, that you only excite stuff which is very close to your surface of your glass cover slide, and you don't have much excitation deep into your sample. So for surface specific detection, this is perfect. And then people came up with the idea to take several images with different incidence angles of your excitation wave. And as you can see, then you have different here penetration depth of your excitation intensity into your sample. And by taking different images and then simply watching how, let's say, an emitter which is placed here at 200 nanometer or 100 nanometer away from your surface, how the intensity is changed for this emitter, you can then deduce how deep the emitter is in your sample. So this is a very nice idea, but of course, in this case, you have to take several images for getting then one number. This is the distance of your fluorescent entity from the surface. This is a near field effect, because what we see here is the near field, the evanescent field, which is generated by this kind of supercritical incidence excitation. There's also a near field when you look at emission. That's a little bit more complicated, so give me a few seconds to explain it. So most of the fluorescence dyes that we use in fluorescence microscopy are perfectly described as an electric dipole emitter. So you have a positive negative charge. They oscillate here with optical frequencies and orientation, the dipole orientation. And when you put something like that into Maxwell's equations, then the result is this famous Hertz dipole emitter. So what you see here is in the very middle, there's this oscillating dipole, and this is the electric field around it. And what you can see is several interesting information. The first one is that the emission is very anisotropic. So there is basically no far field emission along the vertical axis, so along the axis of your dipole. There's maximum emission in the horizontal plane here, and in between is the sine squared theta law. So depending on the angle you are looking on your dipole. The second thing is also there is no far field emission here along the dipole axis. Very close to the dipole you see something. There is a non-negligible, there is non-zero electric field. This is the near field, which is normally not participating in the far field emission, but which becomes very important later. So the fun starts when you take your emitter and you place them very close to an interface with a refractive index jump. Let's say we have here something like air or water. Here's our single molecule close to the surface. Here's a glass surface. Here's glass half space. So what is now happening is the emission of your dipole, you can somehow represent it as something like a superposition of plane waves, is hitting the surface, is partially reflected back. And this back reflected field, so improbable it sounds, can back react on the emission of your dipole here and is modulating its own emission. And the first thing, which is quite important, which is modulated and changed, is the angular distribution of emission. So these two animations now show this is a dipole, a single fluorescent molecule, a dipole in air on a glass surface. And what I'm doing here in the left animation, I simply change the orientation of this dipole with respect to the surface. So from parallel to the surface to vertical to the surface. In the right animation, the dipole is staying in the surface with its orientation, but is rotating with its axis around the optical axis or vertical axis here. And what you see then here is the angular distribution of emission. It means how much light, on average, your emitter is emitting into a given direction. The funny thing is that you can directly visualize this kind of very complex angular distribution of emission when looking on such an emitter with defocused optics. So what is shown here then on the bottom is a model calculation, what you would expect to see with a defocused microscope when you collect the light from such an emitter and image on your CCD. And indeed, this is one of the most beautiful experiments from my colleague Hiroshi Ushi. He is a professor in Leuven. What you see here are exactly these patterns. What we have done here, we have immobilized the molecules, fluorescent molecules, into a very thin polymer film, so they cannot diffuse away anymore, they are immobile, but they can still rotate. And when you look closely, then you see exactly the patterns I have shown you before in the model calculations, and the changing patterns, the dynamics, is the rotational diffusion of your molecules. So we have used this method, this kind of defocused imaging of single molecules in the past, for basically directly seeing the three-dimensional orientations of molecules, and also for different applications in biophysics or in material science. And we have seen a few days ago a fantastic talk by Sandrine Levesque-Faure, who is using this idea to also then to measure the distance of a molecule from a surface. The idea is, when you again look here at the cross-section of the angular distribution of a dipole emitter directly on a glass surface here, then you see that you have this cusp-like emission into the glass substrate. And yeah, there are two parts. This is the so-called anacritical angle fluorescence. So this is here the critical angle of total internal reflection, what you see here. You have the anacritical angle fluorescence, and you have the supercritical angle fluorescence. And the supercritical angle fluorescence, this red area here, is extremely sensitively depending on the distance of your emitter from the surface. When you remove your emitter from the surface, then all the emission here in this red region goes away. And this is shown here. This is the ratio of the supercritical angle fluorescence with respect to the total emission into the glass half space. And you see, okay, the farther I'm away, the less I see supercritical angle fluorescence. And you can use this information again simply by looking at the intensity here in this angular region and this angular region. You can use this information for getting, again, the vertical position of a dipole emitter. And this is was very nicely shown in several papers by Sandrine, and she presented 2 days ago fantastic images using this physical principle. But there's also a second property which is modulated when you put your emitter close to a surface, especially when the surface is covered with a very thin metal film. Again, the physics is very similar. Now, of course, the reflectivity and so on is much more pronounced with a metal film on it. And what you now see is when you come from far away here, from solution, and you approach the silver film surface, what you see is that the excited state lifetime of your molecule is also modulated heavily. Excited state lifetime is when you excite your molecule with light, then it resides in excited states a few nanoseconds before coming back on average, also coming back to the ground state. And this is the fluorescence lifetime. And what you see here is the modulation when your molecule approaches from far away to the surface. This oscillation here is exactly the self-interference of your molecule with its own back-reflected emission. But when you come very close, then here the near-field coupling sets in, so all the near-field components can be reflected, but this is a much stronger interaction, and this leads then to a complete quenching of fluorescence when you come very close to the metal surface, and also the lifetime breaks down. And of course, again, it is dependent on the orientation of your dipole. Fortunately enough for us, in most applications, you will see later, our dyes, even if they are bound to some protein of interest or whatever, is still flexible enough that the dye molecules can rotate, so that we basically only observe then something like an average of these two curves. So why is it now interesting, this kind of spectroscopy for imaging? So at that time, then 10 years ago, we had the idea, okay, maybe we can use this very strong dependency of the lifetime on a distance for doing imaging. So the idea was the following: let's say we have a cell here, we want to map the topography of the basal membrane with a very high axial resolution, much better than a normal microscope would provide. So what we do is we are labeling the plasma membrane with a plasma membrane labeling dye. You can buy them commercially. And then we put our cell not on a bare glass slide, but we have a very small gold film here in between. Very thin means something like 10 nanometer. So it's basically transparent. When you look on it, you barely see the gold film. But this presence of 10 nanometer gold is enough to induce this very strong modulation of the fluorescence lifetime. And then what you need second, you need a fluorescence lifetime imaging microscope. In our case, we take a a relatively standard confocal microscope, but we have as a pulse excitation laser with something like 40 megahertz excitation rate, and then we do single photon counting with a very fast detector here, and then we can measure the fluorescence lifetime. This is the conventional setup. So this is what you need. So you need this kind of gold film, which is easy to make, and then you need this film microscope for measuring the lifetime. And then you can take an image here, and you get two informations. You get the intensity, of course, the fluorescence image intensity, but also the fluorescence lifetime at each spot. And then, because this physics is very well understood here, this is classical physics which was first measured extensively 50 years ago by Drexhager and Kuhn. So because this is very well understood, we can then use the lifetime information and then back calculate the distance from the surface. So we did it, and this was the very first publication then many years ago. And what you see here is now the basal membrane of a cell fluorescently labeled, and the color here now gives the distance here from the surface. And as you can see now, obviously the resolution here, the axial resolution we have, is now much, much better than we would normally have in a normal microscope, which gives you something like 500 nanometer axial resolution. Of course, the lateral resolution here is still the normal lateral resolution of the RB limit, because there is nothing super resolution along the lateral direction here. And we use this method and also to map the mean distance of different cell types. You see here 3 different cell types: NECK2 cell, MDE, MB231, and E50, also 549, whatever it means. And you see that different cells, they keep different distance from the surface. The good thing was that this was already measured many years ago by much more complex interferometric methods, and we find a very good agreement with our method. Of course, the question is, how good can we go? I mean, how good is this method? So what we did some kind of a reference measurement. So we took a glass light, we have here the gold film, then we put here a spacer of a dielectric, silicon dioxide, with a very well-defined, well-known thickness. And then here a super thin film with fluorescent molecules. And then we can make basically a fake experiment. We can simply focus here, we measure lifetime, and then we simply try to get then the axial resolution back. And we do it many times to get an error margin for the measurement. And this is the result here. What you see is the fluorescence intensity and 1,000 counts units here. And this is the axial resolution simply by repeating the experiment many times and looking at the variance. And what you see also is the square root law, which is basically the same thing as you also have in other typical methods when you simply count single photons. So it means the accuracy goes down here like a square root law with the number of photons, but typically we have with a good sample, we have something like 5 to 10,000 photons per lifetime measurement, and this translates into something like 2 to 4 nanometer axial resolution. So we were quite happy, so then we started to use this method. One of the very first applications, it was a collaboration with the Andreas Janshoff lab in Göttingen, and they are interested in this epithelial to mes and chimal transition in cells, and we looked at the reorganization of the actin cytoskeleton upon this transition, and this is here in time. The good thing is you can make these measurements with live cells very easy, so it's live cell compatible. And you see that with this transition here from this one state to the other state, we see something like a 20 nanometer elevation of the cytoskeleton, which is then moving back again after the transition. I mean, this kind of very subtle changes in height would be very hard to see otherwise. Then the next very, popular application was the focal adhesion complex. Here we wanted then to co-localize 2 different units, 2 different proteins. So we did then dual color metal-induced energy transfer. And we labeled actin and vinculin with 2 different dyes, and then again did this metal-induced energy transfer business. And what you see here is again then the intensities and the lifetimes, and this is already translated here into a height scale. And then when you, for example, then pick one filament here in the focal adhesion complex, you can simply move along the filament, and then you can trace the height of these filaments with respect to the surface. And then you see, for example, here that the actin filaments, the actin bundles is always slightly higher than the vinculin bundles here. And also you see this very shallow angle here. When you look here, this is something like 9 micrometers, and we see a height change of only something like 20 nanometers. So the method is really very sensitive. And one of my favorite applications was this here, again, a dual color experiment where we looked at the nuclear envelope. I mean, you, as most of you are biologists, you know very well that the nucleus is not a single bilayer, it's a double bilayer structure. It has an inner and outer envelope of the nucleus, and we wanted to measure the distance between these two membranes. So what we did, we again labeled the inner membrane with one dye and the outer membrane here, nuclear pore complex, the protein with another dye. And then again did this mite business, and the final result is shown here. So what you see here is the result for 6 different cells, and another 6 different cells, and we find the distance here between inner and outer envelope of something like 35 nanometers consistently. And what is super important, this is here called a normal sample and the switch sample, that we switched here in these cells, we switched the order of the dyes. So here the red dye was outside and the blue dye was inside, and here it was the opposite, and we find exactly the same number, which is super important because it also tells us that our method does not have any systematic bias. It does not only give you relative numbers, it gives you really absolute numbers in the distances. Okay, so then the question was, okay, yeah, when we have this metal on our cover slide, of course, we have light losses. Yeah, we cannot avoid it because metal is absorbing light. So we are losing something like 30% of the total emission of the fluorophores. So how sensitive is it? How far can we go in sensitivity? So then my, at the time, student Narayan, which is now a postdoc in Oxford, he then made these fantastic experiments that he used then our metal-induced energy transfer experiments for single molecules. So he put very sparse concentration single molecules here in air on a silicon dioxide layer and then try to image them. And the result is shown here. And this image has several important formations. What you see here are 4 different experiments for 4 different silicon dioxide spacer thickness values, 20, 30, 40, 50. And the first thing what is important is that you see that the signal-to-noise ratio here for our single molecule imaging is really excellent. So although we are losing a bit of the fluorescence, we still have a fantastic signal-to-noise ratio that we can easily see single molecules in these experiments. And the second thing is the color here is indicating the lifetime. And you can see that even 10 nanometers here, change in lifetime is tremendously changing, as in height is tremendously changing in lifetime. And of course, we can also do some statistics, and then we can simply histogram the different lifetimes for the 4 different samples here. And the width again gives us then an estimate how accurate the method is. And we again find here something like 2.5 nanometers for roughly 8 to 10,000 photons per molecule until bleaching on average. So then we said, okay, man, this is fantastic. So maybe we can use now the method not simply to localize some molecules on some artificial samples, but to do something like a co-localization. It would be great to measure distances, let's say macromolecules, with this idea. So what we did, this was a collaboration with Philipp Tinnefeld from the LMU in Munich. They made for us these fantastic so-called DNA origami rockets. This is a complex 3-dimensional structure made out of DNA. And you can place them in a very oriented fashion onto a surface with binding sites here. And on this origami structure, we had then 3 binding sites for fluorophores with a very well-known distance from the surface, so 26, 58, and 82 nanometers. And then we focus on one of these complexes, and we measure then the fluorescence lifetime and intensity. And what you see in the intensity trace, of course, then you have this very nice stepwise bleaching. Yeah, so the first dye is bleaching, the second dye is bleaching, the third dye is bleaching, this is in the background. And then you can go back in your measurement, and then you take only these data, and you get the first lifetime, and then you go back here, then you keep this lifetime fixed, then you determine the second lifetime, then you go back to the beginning, and you keep the lifetime from here and here fixed, and then you fit the third lifetime. In this way, you can then sequentially get the 3 lifetimes for the 3 different dyes, which gives you then the height of these different molecules. And indeed, this is a good message. We find then back the height values that we knew should be there. But we also found that our height values are slightly a little bit smaller than the design values, and it was then indeed confirmed also by AFM that these DNA origami structures, they can show some small shrinking with respect to the computer calculated design. That was very nice, but then we wanted to go so fast. So I mean, of course, what we used so far was a relatively slow piezo stage scanning microscope, so we could only take images, one image per minute roughly. It was enough for the cell motion in the beginning I have shown you, but when you want to look at fast processes, this is not enough. So then what we did, We changed our microscope. We inserted here a fast scan unit from PicoQuant to FLIMBee, which now allows us to take images with something like 30 frames per second. And this is simply an example that it very nicely works. You see toluene labeled as a cells. This is the intensity image. Here's the fluorescence lifetime image. And the good thing now is that we can then also use this now for looking at very fast cellular processes. This was a collaboration with our colleague Sarah Köster in Göttingen. She's interested in this dynamics of blood platelets. When they settle down to a surface and they start to spread extremely fast, and they also rearrange their cytoskeleton. So what you see here is the actin network labeling, and here's the intensity, here's the lifetime already converted into a height image. This is the height image from the lifetime. And you can again see then when the platelet lands on the surface and starts to spread, you see a tremendous reorganization here of the actin cytoskeleton. It's coming down very close to the surface, and the platelet becomes very thin. So that was good. And then the next stage was, okay, when we are so fast with scan images, maybe we can do the trick what people normally do in single molecule localization microscopy. And this is shown here on the next slide. So this is then, we use then now our fast scan unit, and we then process the data, the images, in the same way what people normally do in wide field microscopy for single molecule localization microscopy, like STORM or PAINT or PALM. So what you see here now, this is scan images. These are very fast scan images, and you see then these single molecules here blinking, also lighting up and going down into dark state again. And this was two different samples. This is again, here's a toluene network, and here this is a chromatin. So we labeled here chromatin. This is a PAINT measurement. The good thing now is with this kind of scanning single molecule localization microscopy, you can also now measure the lifetime simultaneously. So this is a fluorescence lifetime SMLM method. And here, for example, for this sample, we then see a very nice homogeneous lifetime distribution of 1.5 nanoseconds, and here for this other labeling, it's 3.4 nanoseconds. Now, of course, you can imagine what comes next. Now we have the lifetime, we can do single molecule localization. Let's combine it with MIT, with metal-induced energy transfer. And this is super recent data, not yet published. So what you see here again is toluenes labeled with fluorescent dye, and this is already the height image where we converted the lifetime for each single molecule localization into a height value. And then when we take here cross sections, for example, here one cross section, here's another cross section, then I think we already can really discern the hollow structure of these microtubules. Don't be confused that the diameter looks much bigger than what you would now from 25 nanometers, because we have, of course, here antibody labeling. So then the real distance of the dyes is bigger than the physical diameter of the microtubules. So this is, I think, then the next step to go. As I said, this is super recent data, and we are now in the way to optimize the method. Then, of course, we wanted to also to know, okay, how good can we come down with our X resolution? As I said, when we have single molecules and only single molecules at one specific position, then we get something like 2.5 nanometers. And this then was somehow encouraging us to think about, okay, maybe we can even then measure something like the thickness of a single bilayer. Yeah, you have a single bilayer, you have these fatty acids, you have these head groups, you label the head groups with a fluorescent dye, you put them on a metal, and then you expect, okay, of course here, because the dye is closer to the metal than here, that the lifetime here for this dye is longer than the lifetime here. However, it was then a hard lesson to learn. When you have many dyes here in such a sample, then it becomes really tricky, because the lifetime difference from here to here with gold is only in the order of 100 picoseconds. Yeah? Whereas if you only have a single molecule which is immobile, you can still somehow get this accuracy. When you have many dyes here, like let's say a densely labeled bilayer, and you have them both at the same time in your detection volume, this becomes really tricky. So we had to think about some alternative. Maybe we can modify the method to get this higher accuracy. And the idea came from colleagues in the Netherlands, Professor Hogenboom, who published in a very nice paper which was showing that also graphene, this wonder material graphene, which won the Nobel Prize a few years ago, also has this quenching property. Of course, it's not a metal anymore. The graphene acts like a super big extended acceptor molecule, let's say. This is more closely now here to further resonance energy transfer, where you have here the donor, and then this graphene sheet is the acceptor for your emission. But the good thing is you also see the same kind of quenching as with metal, but now on a much, much smaller length scale here. So the quenching now is happening over something like 30 nanometers. And this is again showing you for different orientations of the die. So the red curve is for die orientation parallel to the surface. This is for the orientation blue for the orientation vertical, also orthogonal to the surface. And also what you see is that these curves also depend on the quantum yield. I didn't mention it so far, but yeah, we have to know the quantum yield of the die. But then if we know it, then we can calculate these curves very exactly. But the very steepness now here over the very small distance was then encouraging us to try our bilayer experiment. Before that, again, we did some calibration. This is shown here. We put it here on 10 now, very small distance changes here, 10, 15, 20 nanometer. What you see now here are single molecule fluorescence lifetime decay curves. So this is why they are so noisy. And you can see instantly by shifting the molecule only by 5 nanometers away, you have a huge change in the lifetime. So that was good news for us. So then we did the real experiment. We prepared supported lipid bilayers on the surface, but now with graphene as the quencher. We label them on both sides with a really high concentration of dyes. It's not a single molecule experiment anymore, so that's now a high labeling density experiment. And then we measured the lifetimes. And this is now a relatively complex graph. So let's first focus on the left side. What you see here are now lifetime values, and you have these two distributions, which are the bi-exponential fit per measurement. So what we do is we simply focus on the bilayer, we measure fluorescence decay curves, and then we fit them with a bi-exponential fit. And these two values here, the long lifetime, the short lifetime, are the result of this bi-exponential fit. And of course, when you do it many, many times, then you will see that, of course, each fit gives you some kind of error variation here. And this is the width of the distribution, what you see when you measure again and again and fit again and again. And what you see now on the x-axis is how these kind of error, the variance here of these two values depends on the number of photons you have collected in one measurement. And what you see now when you have beyond something like 1 million photons per measurement, then this variance becomes basically as small as you want. This is then the shot noise again. Also you cannot get much smaller than that. This is simply the resolution of our TC-SPC system. Then we can convert these two different lifetimes into a distance from the graphene. This is shown here, and you may wonder why we have a gap here, because here already the presence of the lipid bilayer makes a difference. Yeah, these dyes here with the short lifetime, they are below the lipid bilayer. These dyes here with the long lifetime are above the lipid bilayer, and this is already inducing the gap here. So we have already taken this into account, and then we can, of course, calculate distances. So what you now have here, two different measurements for DOPC lipids or DLPC lipids, and then here it's the bottom top distance, and most interesting number is the difference. So here for the DOPC we see something like 5.5 nanometer, DLPC 404 nanometer. And then of course we also compared it with classical measurements from small angle X-ray scattering. Yeah, this is much more complex method, but people have done it and we can compare it. And it's a perfect agreement. Also the numbers are different because here of course what we measure is the distance between fluorophores, so which are dangling outside of the bilayer on the head groups, whereas what they measure are really the head group to head group distance. Yeah, that's why this is a really very nice agreement. What was also surprising for us that we see a relatively large gap between the glass surface and the bottom of the bilayer in the order of 1.5 to 2 nanometer. And in the beginning we were thinking, oh, maybe this is some mistake, but again it was also confirmed by the Sachs measurements that shows you that even support bilayers have a relatively thick water layer beneath them. Then the next project, which was just published now, was then to use this extremely nice high accuracy to look at something like mitochondrial membrane organization. This is again now a dual color measurement. So what we did, we took mitochondria, we isolated them, we put them here on our substrate, silica graphene glass. We label the inside of the membrane here, the inner membrane of the mitochondria with one dye, the outer membrane of the mitochondria with another dye, and then and it was the interesting part. What we did is we measured these distances, and the mean distances we see between the two bilayer systems in the resting state. So when the mitochondria don't get any buffer where they start to be activated and to generate ATP, and then we added the respiration buffer and activated them, and then measured again. And the interesting thing is that indeed we see a difference. So this is the bottom difference here on the left side. This is the bottom difference for the outer bilayer of the mitochondria to the graphene surface, to the surface, in the active and the resting state, and you don't see a difference. And this makes sense because, I mean, the mitochondria, they are lying on the surface. You wouldn't expect that they change their height above the surface. However, for the inner membrane, we see a clear change of something like 2 to 3 nanometer between the resting state and the active state, and we think this is due to the change of osmolality and osmotic pressure, which is somehow flattening out the inner membrane and brings it closer to the outer membrane. And I would say something like that you can barely do, as you see, with conventional microscopy, and also not with electron microscopy so easily because you cannot do this kind of activation of native mitochondria in a vacuum. Okay, last but not least, my very last slides. The next step, we wanted to see not only static information, so that we see some kind of thickness changes, whatever. We asked, okay, can we see even dynamics? And what we want, when they're looking at, again, this is a very new results, not yet published. We were interested in this cholesterol flip-flop. So we have a bilayer with cholesterol in the bilayer, and it is known from literature that then there is a chance of a flip-flop, so that one of these cholesterol molecules can flip from one leaflet into the other leaflet. But to measure this kind of flip-flop dynamics is very complicated. So again, what we did, we prepared supported lipid bilayer, we added then specific amount of cholesterol with a fluorescent label on it. Then we had again our graphene sample here, and then we measured basically now again the fluorescence signal. What we then see is in the fluorescence lifetime, indeed, we see a bi-exponential decay, which you would expect because you have these two leaflets, you have two different distances, and in the bottom, in the bottom leaflet, then we observe for the dilabeled cholesterol 2.15 nanoseconds, and for the top it's 3.64 nanoseconds. This is good. So we have a very nice difference in a lifetime between the two leaflets, but how can we now measure dynamics? How can we see the flip-flop? This is not so trivial. So what we did here, we then simply took one method we developed many years ago, which is called fluorescence lifetime correlation spectroscopy. So I hope that most of you are familiar with fluorescence correlation spectroscopy, where we are measuring a fluorescent signal and then do a correlation analysis, for example, for measuring the diffusion of stuff through the focus of our confocal microscope. So what we did, we extended the method in the following way. We also used the lifetime information. So we have a system where we can measure the intensity fluctuations, but also lifetime. And let's say you have a system, this is only here some schematic, where we have a system consisting of two components, A and B, and A has a short lifetime and B has a long lifetime, and the green curve is simply the summary lifetime of the mixture. So what we can then do is by using a very clever and tricky filter method, I will not go too much into technical details here, we can then calculate not only FCS curves, which is shown here, but we can calculate FCS curves, as fluorescence correlation curves, in a lifetime specific manner. For example, we can somehow filter only the long lifetime photons and then get, let's say here, the red fluorescence correlation curve, which is, for example, a smaller molecule faster diffusing. Or we can only focus on the short lifetime photons and then can calculate the corresponding correlation curve for the long lifetime, short lifetime photons. This is, for example, the blue curve, which is then slower diffusing. So why is this now important for our flip-flop thing? Because we use now exactly this flip-flop, we use exactly this fluorescence lifetime correlation spectroscopy in our experiments with the labeled cholesterol labeled bilayers. And what we now did is we exactly use the lifetime for filtering and for calculating lifetime-specific correlation curves. And again, what you have seen before, this is the one-to-one fluorescence correlation curve for the long lifetime. This is the 2-to-2 is the short-to-short photon correlation lifetime, so correlation curve for the short lifetime. But what we also can do, and this is not a really interesting part, we can also do cross-correlation. So we can handle the lifetime like a two-color measurement, a two-color cross-correlation measurement. So we can calculate the correlation function between long lifetime to short lifetime photons or short lifetime to long lifetime photons. So what does it mean physically? It means, let's say we start with the cholesterol in the top leaflet, then it's a long lifetime, and then during the observation time, it does a flip-flop, and the lifetime is getting to shorter time. So then you would expect in this cross correlation this kind of anti-correlation. This is exactly what we see, and the characteristic time here of this anti-correlation is the flip-flop time you see here in our experiment is in the order of a milliseconds time range. So indeed, we can now use also MIT or graphene-induced energy transfer, as we call it, not only for static measurements, but also for looking at very fast dynamics in systems. So with this, I'm already at the end of my talk here. I want only to mention that we have developed an open source software you can download from our website. Also did some kind of correction for fast lifetime film imaging. This is all publicly available, so if you have any question if you want to do something like that, simply ask us. We are happy to support you in any way. Of course, I have to thank my whole group in Göttingen, also past and present. So what you see here is not only now, that's also from past years. And especially Anna and Alexei, who started this MIT business in Narayan. He even wrote a book, highly recommended. There's a Springer book from Narayan about metal-induced energy transfer. Arinda now is a postdoc with Markus Sauer in Würzburg. Daya did a lot of the software development. Sebastian was fantastic with this co-localization. He is now with the Balsarotti lab in Vienna. Akshita did the spy layer stuff with Arinda. And this is my present co-workers, which are continuing this MIT business. And last but not least, I have also the pleasure to simply make some advertisement for a new journal. I have the big honor to be the editor-in-chief of this new journal, Biophysical Reports from the Biophysical Society for Biophysics, for advanced biophysics stuff, and have a look. It's gold open access, so it's freely, there's no fees for reading it, and I would be happy if you submit something in the future from your research. So thank you for your attention. I hope it was not too fast or too long. Yeah.
Okay, thank you very much for this very interesting talk. Do we have questions?
Yeah, fantastic talk. Thank you, Jörg. I have two questions. One is about, I see that you use a lot dyes for this technique, but how about fluorescent proteins? Have you tried?
Excellent question. I mean, for the mitochondria, one of the dyes was M-cerulin, so a fluorescent protein. But by the way, very good question, because we are now preparing a big manuscript where we did exactly this studying that we can also use MIT with fluorescent proteins in living cells and so on, which is of course super important for life cell
exactly.
work. Yeah.
And another question is about this flip-flop dynamics. Would it be technically possible to do it at the single molecule level, to do imaging and to see?
Also not with this fast dynamics, because here we have a flip-flop dynamics of a millisecond. So that would mean that you would need to collect within 1 millisecond enough photons to really get a lifetime with 10%, 5% accuracy.
But maybe you can do statistics on intensity, because you probably have a huge difference in the intensity of the...
Yeah, this is a good idea. We did this with something like, let's say, a single DNA strand which are flopping around, and yeah, you can get then dynamics. But again, we do a correlation analysis. Yeah, but of course you can use for this kind of analysis correlation, you can use the lifetime, what we did here for the cholesterol, but you could also basically do intensity correlation, and then you would see in the corresponding time scale simply a partial correlation decay. Okay. However, then often you would then come into the same realm as photophysics, yeah, and then you don't know, is it now triple state dynamics or is it the real stuff you want to see? So the FSCS is good because it's really specific. No? It's only when the lifetime is changing you see a signal.
Yeah, okay, thank you.
Thanks for the very nice talk. I have a couple of questions about the MIT for localization microscopy. What are the potential for using camera-based detection? And the second question is, did you think about, because the lifetime seems to be a really independent orthogonal measure, can you combine this with things like shaped, like PSF engineering or patterned illumination in order to further increase the information?
Also first, let's say the second question first. Yeah, I mean, sure, you can combine it with, for example, PSF engineering for getting better precision, whatever, because it's, as you said, it's orthogonal information. Ne? The first question was camera-based. Yeah, we have now in our lab a very nice lifetime, as a single photon lifetime camera, yeah, which works really nicely. This is, I didn't show it here because, you know, it's very brand new. And it has one caveat, it's super expensive. Yeah? So this is really the downside. The funny thing is the camera has a quantum efficiency of only something like 8%. And when we first looked into that, we said, okay, this is hopeless for single molecules. Then we checked it out and it works fantastically well because it is zero background. This camera has 0.0 background. Yeah? So that even with 8% quantum efficiency, we can do with nearly the same accuracy, single molecule localization and everything what I have shown here with the scanning system. Yeah, but I mean the downside is it's expensive, and they told us it will not get much cheaper. Yeah, because the technology is the limit. Yeah.
Thank you, Jörg, for the nice talk. In the DNA origami experiment, do you have an idea of the orientation of the dies attached to the origamis, and does it play a role in the...
Also you have a link.
Free.
Yeah, it's a linker. And it's a linker with several carbon atoms, and the die is really rotating. So we also check, normally we also check the orientation by anisotropy measurements, because the orientation, of course, is critical. When the orientation is not free, then it will bias the measurement. So we have to check it. For some of the membrane, for example, we use the your die, Andre, and for some of the dies we see that's a preferred orientation with respect to the bilayer, and we take this into account. Yeah, so for example, the bilayer measurements with the graphene, the thickness, we took there the orientation of the die into account. Yeah.
Thank you for the amazing talk. Since you're translating lifetime to Z, I was wondering, what are the confounding factors that could cause a change in lifetime that are not Z? And
yeah, it is a very important question. So for all the cell measurements, for example, we make sure by control measurements without metal or without graphene that the lifetime when you don't have mite is really homogeneous and not changing by everything. And many dyes don't show this nice homogeneity, so you have to check that in advance. So for each cell, this is really the bottleneck. So for each cell sample, for each cellular question we are studying, we have to check that the dyes we are using don't show any weird behavior without the mite effect. This you have to do, yeah.
Thanks.
Great talk, thanks, Jörg. I was wondering, did you investigate whether, like, how the proximity to a metal actually changes or not the dye's photophysics in terms of, like, things like triplet state intersystem crossing or even like photo bleaching yield? It
changed, of course, yeah. For example, with the mite, what we see is really funny. The closer your dye is to the surface, to the metal, the dimmer it is. There's only lifetime, also the intensity goes down. But the good thing is, because the excitancy lifetime is shortened, also the chances for photo bleaching are going down. And what we see is the funny effect, and in the end we get the same number of photons roughly until bleaching from all heights as without a metal. Yeah, that's good for us. So the faster you de-excite a molecule, the less chances it can make some bad things. Yeah.
Thanks.
Thank you for the wonderful talk. I have a question concerning your measurement in lipid bilayers, outer leaflet and inner leaflet. Do you have some problems with homothread in this case?
No, I mean the label density is never so high. So I mean, we are never coming so close that these dyes are coming in homo-thread distance. So we are using well-labeled bilayer, but not so highly labeled. Yeah. So we never saw any bad effects, homo-thread or whatever.
Okay, if we don't have any more questions, we'll move on to the next talk. Thank you again.
It's my pleasure to now introduce Dr. Collinson. She's an electron microscopist with a background in microbiology and cell biology. She received her PhD in medical microbiology in 1998 and then conducted research with Professor Colin Hopkins at the MRC lab for molecular cell biology at UCL and then at Imperial College, where she investigated membrane trafficking pathways in lysosome-related organelles. She then ran a series of biological electron microscopy facilities since 2004 at UCL and then at the Cancer Research UK London Research Institute. And since 2015, she leads the electron microscopy science technology platform at the Francis Crick Institute in London. Can you hear us?
Oh, so then hi everyone. Can you see my video okay?
Yes.
Yeah, great. Okay, so thank you very much for having me speak, and I'm very sorry that I can't be with you over there. It's feeling very cold and dark in London right now, so I think I'm missing out. Today I'm going to talk to you about approaching quantitative correlative microscopy, and I'm sure you've all had a long day with lots of intense work on the microscope, so I'll try and make it entertaining. So what is correlative light and electron microscopy? Well, it's a workflow rather than a microscope. And using this workflow, we can find a fluorescent protein which is expressed in cells or tissues. And then we might image the sample using bright field light microscopy. So this is a cover slip with neurons growing on it. And then we link that light microscopy data to another imaging modality like electron microscopy. And that means that we can track exactly the same area and image it at much higher resolution. So here we can see that our protein is localized to a synaptic vesicle, which is communicating across the synapse. So that means that we get the benefits of fluorescence microscopy, so we can see dynamics in live cells, and we can see molecules that have been labeled, but we can also see the structure of the sample, so we can see the context of the underlying cells and tissues. And there are different types of correlative workflow, including cryo, volume, and precision. And today I'm going to talk about two of these areas, concentrating on volume, clem, and high precision clem. So starting with volume correlative light in EM, we really go back to 1986 to this incredible publication nicknamed The Mind of the Worm. So this was Sidney Brenner's team looking at C. elegans and mapping all of the neuronal connections. And that was an effort that took 10 years using a technique called serial section transmission electron microscopy. So in this technique, you have your cells or your tissues embedded in a resin block, and you then cut serial ultra-thin sections, one after the other, using a diamond knife onto a boat of water, and you pick those up onto grids. So this is a Formvar coated 3mm transmission EM grid. And then you image that in the transmission electron microscope and acquire the images. And then in a post-processing step, you stack them all up together to make a 3D volume. So back then, there wasn't the computer processing to be able to do that in silico. And so there are all sorts of ways of visualizing the data in 3D, including cutting out neurons using cardboard and sticking them together so you could actually see the neurons moving through volumes. Luckily, things have progressed quite a lot since then. So nowadays, it doesn't take us 10 years to collect this kind of data. It we can do it in weeks. And there's been an explosion in the number of volume electron microscopy techniques that are used in the biosciences. So we also use serial electron tomography, where we cut slightly thicker sections and then use tilt tomography to reconstruct a much higher resolution through the volume. And with progress in scanning electron microscopes in the columns, the sources and the detectors, there are now techniques that we call block face techniques, like serial block face SEM and focused ion beam SEM, where the same sample is put into the chamber of the scanning electron microscope. And then instead of cutting on a diamond knife with a boat of water, you either use a diamond knife in situ inside the scanning electron microscope for serial block face, or a gallium ion beam in focused ion beam to do your cutting for you. And because this is done inside the scanning electron microscope, it can be automated. So you cut a section, discard it, and then image the sample surface, and cut an image, and you build up a stack of images through the volume. There's also a newer technique called array tomography, which has a similar sample preparation method to the original serial section TEM. So here again, sections are cut onto a boat of water, but they're now picked up either manually or automatically onto a large silicon wafer. So this could be 4 or 6 inches, so we can now fit thousands of sections onto one substrate. And then those are imaged in a scanning electron microscope. And unlike the block face techniques, array tomography is non-destructive, so you can keep a library of these wafers through the volume and you can image them multiple times. And now there are advances in automation for serial TEM imaging. So transmission EM will always beat scanning electron microscopes on resolution. So if you really need membrane bilayer resolution, but you want volume, nowadays you might use the grid tape transmission EM or the Tempka transmission EM. And similarly, there are also advances in focused ion beam, even though this is a much newer technique to biology. So we're already seeing gas cluster ion beams and plasma fibs, which can mill much faster and much larger volumes than the gallium ion beam, and laser milling, which can help us to quickly prepare faces for high resolution imaging. And then over in the scanning electron microscope domain, we have advances in array tomography with multi-beam scanning electron microscopes that have parallel electron beams that can image at the same time. So you can now image over sections that are millimeters in width, but still with 5 nanometer resolution. And a suite of techniques mainly based on X-rays that either image whole tissues with resolution approaching what volume EM can achieve, or allow us to see inside a sample to help us really relocate a region of interest for correlative. So as you can see, volume electron microscopy is actually a huge number of different techniques that all deliver similar data. And the field has progressed now so that, looking at the mind of the worm back in the 1980s, now we're seeing efforts that have mapped the mind of the fruit fly and zebrafish, and moving towards the mind of the mouse. And there's a really beautiful analogy in this paper of the different scales and the challenges of imaging these systems. So if the challenge or the size of the C. elegans brain is equivalent to the width of one airline seat, then imaging the Drosophila brain is like imaging 6.5 Boeing 747s. And the huge challenge of just imaging the mouse brain, not even going anywhere near a human brain, is like the distance flying from Boston to Lisbon. So even though we're about here in the effort, there's still a very long way to go even to image a single mouse brain. And like all types of microscopy, sample preparation is absolutely key. especially because with electron microscopy, you really are seeing the nanoscale structure of cells. And so if you do anything to your cells that they don't like, then you'll see it in the ultrastructure and you'll get artifacts. So a standard sample preparation for EM starts with either cryo or chemical fixation. I'm going to just talk about chemical fixation today with paraformaldehyde and glutaraldehyde, and that cross-links the proteins in the sample and stabilizes it. We then add heavy metals, so normally osmium tetroxide, which cross-links the lipids in the samples and adds electron density, so it's also stabilizing the sample. But in these volume EM techniques, we have to get extra stain into the sample so that it's conductive in the scanning electron microscope. So we also add uranar salts and lead salts. Then we dehydrate using a solvent, usually ethanol or acetone, and that removes the water, which means that we can then infiltrate with a liquid resin, usually in epoxy, sometimes in methacrylate, and then we polymerize either using heat or ultraviolet light. And once you have a hard block, that protects the sample from the vacuum in the electron microscope and allows us to cut it very thinly. So just looking in a bit more detail at one of these volume electron microscopes, this is a serial block face scanning electron microscope. So we have a standard SEM over here, but we've taken the normal door off and replaced it with a 3-view door from Gatan. And inside, attached to that door, there's a miniaturized ultramicrotome that has a diamond knife that cuts a slice from the surface of the block, and then you image the cells or the tissues using the electron beam. And then you take another cut, discard it, and then you image the surface. So slice, image. And that gives you a video that plays through the volume. So this is a pellet of HeLa cells, and we're scrolling through, and we're imaging at 5 nanometer pixels and cutting at 50 nanometer voxels. So we have 50 nanometer slices. So we have non-isotropic voxels here. You can see the structure. So obviously this movie is binned a lot to be able to show it to you. The original data set's about 500 gigabytes, something like that. And so this kind of data block, which is a few hundred microns, will fill up an entire hard drive. So you can image cells, but you can also image tissues. And on the left here, we can see some mouse liver. So there's a cell nuclei, loss of endoplasmic reticulum because we're in liver. Got a blood vessel up here with a red blood cell. And on the right-hand side, this is mouse retina. So again, we've got some blood vessels looking at endothelial cells. So how do you integrate these volume EM techniques with a correlative workflow? So I'm going to show you an example and then a few other systems that we've used this workflow for. So in this case, the work is done by Marie-Charlotte Demart in my team, and she was collaborating with Nagisa Yoshida in Serge Mostovi's lab in collaboration with Eva Frickel. And they were looking at zebrafish embryos, trying to work out if they would be a good model for studying parasites. And in this case, particularly Toxoplasma, which you can see in green, labeled with GFP, infecting a zebrafish embryo hindbrain. You can also see that they've labeled macrophages in red. So they're looking at the host response to the infection. So the first thing we do is we look at the light microscopy. So that can be any type of light microscopy depending on what you've optimized for your sample. In this case, Nagisa was doing imaging on the entire zebrafish embryo, but then we were also taking vibratome slices through the zebrafish hindbrain and then imaging them with an airy scan. So this is about 100 microns in depth, and you can see individual Toxoplasma parasites with macrophages. So as I said, in some cases we'll then use X-rays, usually as an intermediate step. And with X-rays, like with a hospital CT system, we can see inside the sample non-destructively. And with heavy metal stained soft biological tissue, we can see a lot of ultrastructure. So this scan has a resolution of about 1 micron. It's done on a lab CT system, a Versa 510 from Zeiss. And you can see over here in the 3D model, you can see the eyes and the hindbrain. And if you start slicing through the data, you can even start to see individual toxoplasma parasites. So that gives us 3D position of the region that we're trying to target. And that means that when we go to serial block face SM, we can pick out individual parasites within the volume to image. So in each of these movies, you'll see the parasite appearing in the center here. You can also see that we've captured the entire volume of each parasite. And that's really important because then we can scroll through the different planes and you can start to see features. So the arrows are pointing to host cell mitochondria, which are surrounding the host membrane, which is encasing the parasite. It's the parasitophorus vacuole. We shall call it the PV because it's much easier. And you can also see that in this case, there is one parasite. In the middle case, there is one parasite, but it's in the middle of dividing and you can still see a connection between the two parasites. In the third case, you can scroll up and down through the data and you can see that this is post-division. So then the two parasites have divided and they're no longer linked. And that's one of the reasons why you need 3D for this work. But we can also start to be quantitative. So we can now target multiple parasites within the hindbrain. And in all of these cases, we're seeing a single parasite. In the coral pink border images, they're dividing. And in the purple border images, they've already divided and there's a doublet. So already we know that the toxoplasma parasites can replicate within the zebrafish brain. So there was a lot of imaging experiments done, and one of the important ones was when the reviewer came back and wanted higher resolution. So here we're imaging at 5 nanometer pixel size, 50 nanometer depth. If we move to focused ion beam SEM, where we're cutting with the gallium ion beam, then we can cut at 5 nanometer slice thickness. And that means we have isotropic voxels, and we can turn the data around in silico and look at it in any direction. So again, MC did the ARIScan imaging on 100 micron thick slice from the zebrafish, and then located a macrophage which contained 2 parasites. And that's what's going to appear here. So we're now scrolling through the data, we're at a higher resolution. You can see some features of macrophages like membrane whorls from in the kind of lysosomal compartments, and the 2 parasites appearing and disappearing there. If you look on the left, you can take the 3 ortho slices from the fluorescence microscopy and line them up with 3 ortho slices from the FibSem dataset. So you can see the 2 parasites here. In this plane, you just see one of them, and in this plane, you just see one of them. And the particular questions that the reviewers wanted answered were whether the PV, the vacuole that the parasite is sitting in, is complete, or whether the parasite has managed to break through. And the blue arrows are highlighting that very fine PV membrane, and actually we do see big breaks in that membrane, which you can see in all 3 dimensions and scrolling through the volume. So that FibSem was done by Chris Peddy in the lab. So I hope you can see the value of moving from light to volume electron microscopy and how each modality gives you different information which tells you more about the whole biological question. Just to show you some other ways that the lab have used it. So this is again Marie-Charlotte working with Eva Frickel looking at toxoplasma, but in this case using serial section transmission EM. So we have the fluorescence and then the parasite in individual cells growing on cover slips now. looking at different mutants and different states and whether the PV is intact. Then we have some work from Azumi in the lab. So this is looking at Drosophila nephrocytes, so kind of kidney cells, and particularly looking at endocytic pathways. So here we have different stages of the endocytic pathway labeled. So we have dextran uptake and then RAB7, which is marking the later endosome compartments. And of course, it's much more informative if we look at the movies. So again, this is serial block face SEM. And you can see the strand is taken up into these early compartments, don't complete on later RAB7 compartments. And this was repeated for 6 nephrocytes. It was a large number of samples for correlative. So we generally have a problem where n equals 1, or if you're lucky, n equals 3 in a correlative microscopy experiment, because the workflow takes so long and it's so challenging. And then this is some work from Chris on an even bigger sample. So this is looking at zebrafish intestine and looking at potential glial cells. So those are labeled in green. We also have neurons, I think, labeled in red. So again, you don't really get the full picture until you start looking at the movie. And this is using focused ion beam SEM, but through a very large volume. And if we sat here and watched the whole movie, we would be here all night. So I'm just going to try and drag through. So you can see that by integrating the 2 modalities, then we have cell identity on top of cell structure. And we can start to analyze where these 2 cell types are interacting and what the hallmarks of the cells are. So just coming back briefly to micro-CT. So Anne in the lab is in charge of our micro-CT system and does some development work on it, including looking at how we best stain the samples for imaging. This is zebrafish embryos on the left, and also pushing the resolution limit. So one thing that I find absolutely astounding is that you can embed a pellet of cells or a cover slip with cells on them into resin with heavy metal stains. And if you put them into a micro-CT system, you have enough resolution to see individual cells, but not only that, you can see the nuclei, you can see chromatin patterns, and you can even start to see mitochondria. So that means that we easily have single cell targeting ability using X-ray. We don't always need to target a single cell level, so MCs used the micro-CT to look at brain slices to find putative tumours and to analyse their hallmarks. And Alana has used the micro-CT to localise a really tricky organ called the carotid body in mouse, and to then look at the effects of hypoxia on cells with granules in that body. But we can also include it into some really complicated experiments. So this is some work done with Azumi, and I'm just going to show you a video that demonstrates how you link together fluorescence microscopy, X-ray microscopy, and electron microscopy to answer a research question. This is a collaboration with Suzanne Nolschag, and here we're looking at mouse blood vessels. So this is in the cremaster muscle, and it's a model for inflammation, and it's a mosaic model. So we have different cells sitting next to each other. So we have ATG, 5 knockout cells next to wild types. And the aim was to look at the cell-cell contacts, and in particular autophagy related compartments at these contacts. So we've got wild type connections, wild type knockout and knockout connections in the same tissue. And also to look at immune cells, so neutrophils crossing over between these boundaries. So going back to the technical side, this is going to go quite quickly. So we have beautiful Airiscan imaging of the blood vessels in the cremaster muscle. We're now looking at the embedded piece of tissue, and the red dot is the region of interest. This is now a low resolution micro CT of the muscle with the blood vessels in yellow, which you can see match up with the fluorescence microscopy. So Zumi then trims down the tissue, does a higher resolution micro CT scan, and checks that the region of interest is still there against the light microscopy. So you can then overlay those two data sets and bring them in and out and see where the nuclei are in the individual cells. And that helped her to trim down to the very specific region of interest, just a few hundred microns, to capture the serial block face SEM data. So you go through the serial block face SEM and then segment out that data, use a nucleus map to overlay onto the original fluorescence data. And that's what you're seeing up here. So we have a segmentation of the endothelial cells, and then those were modeled, and you can see flaps in the endothelial cells where you get neutrophils moving across that barrier, and co-localization with the autophagic compartments. So that was a tour de force. Like all of these experiments, they take a lot longer than you hear about in talks. So that, I think, was 2 years' work to get to that point. So I'm now going to move on and talk about high precision correlative workflows very briefly. So I'm just going to mention data first. I've got my slides in the model. So when you have big data, you need to segment it, because if we're looking at a series of 2D images, then we're not very good at understanding what those structures will look like in 3D. And the way we do that is normally to sit in front of each image and to draw around the region of interest. So this is Anne drawing around a nucleus on a touch screen in the lab. And to make a 3D reconstruction of a single nucleus would take about a week, going step by step through each image and drawing around the nucleus and then putting those lines back together. But this is really important because until we can see the structures in 3D, we can't interpret the meaning of them. And we have plenty of publications where we've had to spend months or even years reconstructing here endothelial cells fusing, and here stem cells differentiating, and over here immune cells, to understand what the experiments mean. So it's not sustainable for us to sit in front of the huge quantities of data that we're producing and manually segmenting them. We just don't have enough expert electron microscopists to do it. So Martin Jones, who runs our microscopy prototyping lab, had heard of something called citizen science and teamed up with Chris Lintott, who runs the Zooniverse platform at the University of Oxford, and Helen Spears, who is the biomedical lead for the Zooniverse and now a Chan Zuckerberg Initiative imaging scientist, and built this platform called EtchACell. So if you've got your phones out, it's probably a bit small, but you can hold them up to the QR code. This will come back later if you want to have a go. And the idea is that we feed out every image and we ask the general public to do our segmentation for us. So we show them a central image and then a few images either side for context, and ask them to draw around organelles. And this is actually a really good public engagement tool. So it's been used in various exhibitions at the Crick, and it's being used in schools. And we have feedback from them that it's really good for teaching students about cell biology. So in that first project, looking at the nuclear envelope called EtchACell Going Nuclear, we had more than 6,000 volunteers contributing to the project, with more than 130,000 lines drawn on images. And this is just looking at all of the lines drawn on a single image. And you can see some of them are really good and some of them are not so good. And the reason we have multiple lines on an image is so that we can get some consensus. So hopefully if we give the image to 15 or 30 different people, then if we take an average, then we'll end up with a good line. Actually, it turns out that that step is incredibly difficult, and it took the CRIC scientific computing teams and our students about 2 years to crack it. But once they managed it, you can see that the expert lines are very similar to the aggregated citizen science lines. And we then use this aggregated citizen science data to train a UNet. And the results were really impressive. So if you measure the average house door distance from the machine learning to the expert ground truth, we're less than 20 nanometers away. So this is good enough for our biology. There was one other thing that we had to do, and that was to run our algorithm in all 3 directions in X, Y, and Z, and that filled in any holes we had in the nuclear envelope. And you can see that we are now using the same UNet and training it with citizen science data and expert data in 2 different work streams. So we can now automatically reconstruct mitochondria. And we can do this at scale. So moving from one expert reconstructing 1 nucleus in 1 week, we can now reconstruct many nuclei, and we can start to think about a new science of morphometrics, which is where we generate organelle meshes and we're able to measure their features and compare them in health and disease in a way that hopefully will soon be statistically significant. So this work, like I said, is done by with Amy Strange in our scientific computing team, and her team and an undergraduate student who was with us for a year. And the important thing is, once you have a mesh, then to understand that these surface meshes are different when they're being generated by different software. So if you generate a mesh in iMod, it will look different to a mesh in Fiji. So that doesn't matter if you're just using it as a visualization tool, but it does matter if you're trying to measure features. So we had a second student, Adam, who was with us for the last year, and Adam has designed a workflow in which you condition the mesh, and then it goes through a quality control step to make sure that during the conditioning process, you haven't changed it from the original segmentation. And this, we think, is going to be a critical step in actually making these measurable. So you can get involved. The original nuclear envelope project is closed, but we have 2 projects open at the moment, one in collaboration with Rob Parton and John McGee in Australia and Kerry Thompson in Galway. And this is Etch-a-Cell VR, virtual reality, where we're going to reconstruct an entire cell. And the other is with Jez Carlton and Guy Pearson at the CRIC, and this is working on the endoplasmic reticulum. And this is the most challenging project to date because there's a lot of endoplasmic reticulum in the cell. So one unexpected resource that comes out of this is the benchmark data that we've collected for these segmentation automation projects. And we deposit these in Empire. So Empire is the electron microscopy public image archive, which was set up by EMBL EBI, the European Bioinformatics Institute. So you can navigate to their website and search either for Collinson or for these numbers, and it will bring up these data sets, and you can download them and reuse them. And we've also now moved into collecting benchmark data sets for correlative light and EM to help programmers to access this data and to be able to design better automated solutions for image registration. So finally, coming on to precision. So back in 2014, we published a paper that was inspired by a couple of previous pieces of work from Susan Nixon in 2009 and from Kent MacDonald and Rick Webb in 2011. So in this protocol, we put cells and tissues into a high pressure freezer. So we're fixing them using cryofixation, and we're avoiding chemical fixatives. And then we exchange the vitreous ice for a solvent at cold temperature before the water can recrystallize. And that solvent contains a very low proportion of a nice heavy metal, uranyl acetate, instead of a toxic, nasty, heavy, I mean, they're all toxic, but instead of a nasty heavy metal like osmium tetroxide. And then we bring them back up to room temperature and embed them in a methacrylate resin. So these resins will tolerate a small proportion of water, and they're not particularly autofluorescent. And when you do that, you maintain the fluorescence from proteins like GFP, RFP, M-cherry, CFP, YFP, and you can still see them in the block face, and you can zoom into the block face and see individual cells. So here, this cell is expressing M-cherry H2B in the nucleus and a probe called GFP-C1, which binds to diacylglycerol on membranes, including the nuclear envelope and the endoplasmic reticulum. And we call these samples in-resin fluorescence samples. And that's because the cells are in resin and they're still fluorescent, but they also contain heavy metals, so we'll be able to image them by electron microscopy. So these samples provide some unique challenges, because when we're cutting resin blocks for electron microscopy, the way that we identify the cells and the tissues in the resin block is because they're black, so we can see them under the eyepieces in the ultramicrotome. But when we leave out osmium and we only have uranyl acetate in the sample, then we can't see where the cells anymore, so we can't trim them with a razor blade, and we don't know when to pick up sections. So Martin, again, the lead of our microscopy prototyping team, and Lizzie Brahma and now Wei Guan, built this contraption, which is a miniaturized fluorescence microscope integrated into an ultramicrotome. So here is our sample, here's our diamond knife, and this is our light microscope. And it works like this. So we can shine light from underneath the sample, which means that we can see where the fluorescence is to trim it. And then we can put the sample into the chuck, using a glass knife, start to trim the block. And every time the sample comes up and over, it takes a fluorescence image. So every time there's a section cut, we take an image. And then when you're ready, you can take the glass knife out and exchange it for a diamond knife. So you can see we've swung the light microscope out of the way here. So you do your alignment, and then you bring the fluorescence microscope back in, you align it, and then you start to cut sections. So again, we've got the sample going over the knife, coming back up. You take a fluorescence image, which you can see up here, and then it goes down with the knife again. And eventually, you can move the light microscope out of the way and pick up sections from the boat. So what we have here is fluorescence-guided ultramicrotomy. And there are some interesting byproducts. So what we're effectively doing is serial fluorescence imaging on the block face. And out of that comes something even more interesting. So this is quite difficult to explain. If you're focusing at the block face, then cells that are deep within the block have a kind of point spread function that comes towards you. And when it hits the block face where you're focused, then you see a circle. It's kind of the point spread function. So these circles we can detect using image analysis algorithms. And as you cut away at the sample, and as the cells come closer to the block face, then they become spots and then they disappear when they're cut away. Because these circles become smaller, then we can use them to predict when each object is going to come to the front of the block and when we should collect sections. So this is work done by Martin. Martin came up with this amazing idea and has been working on it with Lizzie, Chris, Wei, and Azumi. This manuscript is just about ready to put out, so we're very excited about it because this means we can now automate the process of collecting fluorescent sections for high resolution CLEM. So once you pick up those sections, you can pick them up onto TEM grids and you can image them in a fluorescence microscope and in an electron microscope and overlay the two images. And what you have now in fluorescence, you have a super resolution image in Z because you've physically cut a section of, say, 100 nanometers. So we're already below the resolution limit that we would get in a standard confocal, even some super resolution microscopes. And you've also removed all of your out of plane fluorescence. So you have really beautifully crisp images. And because you're imaging the fluorescence and the electron image in the same section, then they overlay really beautifully. So here we've got diacyl glycerol localized to the nuclear envelope, to the tips of the Golgi apparatus, and to groups of vesicles in the cytoplasm. You don't have to image them in separate light and electron microscopes. You can image them in an integrated light and electron microscope. And the system we have at the CRIC is the Delmix CCOM. So this is a little bit like the serial block face SEM, as in you take your standard door off the scanning electron microscope and you replace it with this blue door, which is effectively a fluorescence microscope. So it has a couple of laser lines which go through a vacuum port on the door, and then the light microscope objective comes up under the sample for fluorescence imaging, and the electron column comes in above for electron imaging. And you can align the two columns, or the objective with the electron column very finely, because when the electron beam hits the substrate, so this is usually a conductive glass, then it creates a cathodoluminescent signal which you can see with the light microscope objective. So you can put down a pattern and align the two beams to each other. So this is now a different sample. This is a thin section, 200 nanometer section of HeLa cells embedded in resin using our in-resin fluorescence protocol. And the HeLa cells are infected with vaccinia virus. So each of these spots here is a virus particle, and it has YFP labeled in the virus core. So if we zoom into one section, you can see, again, each of these is a virus particle and each of them has YFP. And you can see that the overlay of fluorescence to the electron signal is now very close. So these virus particles are about 200 nanometers, 250 nanometers in diameter. But there's something really interesting about putting these fluorescent proteins into resin. So as many of you will know, if you image this is YFP in a whole cell, and you turn up the laser power and you try and drive it to blink, GFP, YFP, CFP, and others, they don't blink very well, so they're very difficult to do super resolution light microscopy with. But when you embed the same samples into resin and cut them into sections and image them either on a normal fluorescent microscope or inside the vacuum chamber of a electron microscope at 200 pascals of water pressure, water vapor, then they blink like crazy and they carry on blinking, and you can change the vacuum conditions and you can reactivate them. And that means that you can collect 40,000, 60,000 images, and you can use single molecule localization microscopy software like Thunderstorm to reconstruct a super resolution image. And when you do that, so this is now looking at a section using wide field fluorescence in the scanning electron microscope with cells and virus particles. When you do the super resolution reconstruction, this is what you see. And then you can switch directly to the electron microscope and take the EM image and see the underlying cells. So if we zoom in, you can see the wide field image, the super resolution reconstruction, the EM, and then the overlay of the super resolution image onto the underlying cell structure. And what you see now is that the super resolution image has shrunk down the localization to the core of the virus, and working with Susan Cox, we did some analysis on the localization accuracy, which comes out at about 80 nanometers. So we can achieve a three-dimensional localization accuracy of 80 nanometers, but I think we can probably push it in the future towards a 50 nanometer localization accuracy. But there's always a but, and the issue at the moment in this protocol is the contrast compromise. So when the cells are stained, they have one side of the pellet or the tissue which is open to the heavy metal stain, and one side over here which is up against the metal planchette. So you get a gradient of heavy metal stain. So you can see we've got very heavy staining on this side, so we'll have very nice morphology in the electron microscope. And we have very little stain on this side, so the membranes will be difficult to see. And we get a gradient of fluorescence preservation going in the opposite direction. So actually we have more fluorescence preservation where there's less electron stain. So we need to overcome this challenge to be able to apply this to routine biological research questions. And there are different potential solutions. The first is to overexpress your protein of interest, but I'm sure you all know that that's not always a good idea. So this is a kind of endogenous level expression, but at the bottom here we have a darker cell, heavier stain. We still have nice fluorescence because it's overexpressed, but what we've started to do is to wrap up the endoplasmic reticulum membranes. So this is an artifact caused by having too much of the C1 GFP probe in the cell. So it's not a good option generally for sound biological interpretation. And then Chris and Sangeetha Hari from Delmic did a 6-month study trying out all of these different heavy metal stains and rare earth stains to see if we could find something that was compatible with fluorescence preservation and would still give us good electron density. And at the end of all of that, they found out that uranyl acetate is still the only good option. So we came to a bit of a halt on that one. But what is really interesting is the arrival of these new osmium-resistant fluorophores in the field. So these are being developed by labs at Janelia Farm with Lauren Luger, and that's the MEOS 4B probe, and there are now more coming through in dual color. And these are not yet a magic bullet because we're finding that we get about 15% preservation of the fluorescence emission when we add osmium. But I think if we change the embedding protocol or with new generations of these probes, then I think we could be looking at the magic bullet that allows us to conduct all of our experiments with post-embedding CLEM, which means that we can get very high resolution overlays of function to structure. So then we want to do this in whole cells and whole tissues. So now we have to also bring in a volume EM technique, and we're really talking about array tomography here. And what you can see is, whereas I was showing you single sections from vaccinia virus infected cells, now we're seeing serial sections which have been put down onto ITO coated glass and imaged in the fluorescence microscope. And then the same sections are imaged in the electron microscope. So just a cartoon of what we're expecting to get. So to go to volumes, we will acquire serial section single molecule data with serial EM data from the same sections, which we then put together into 3D models of the organelles using our machine learning algorithms to model the nucleus, the endoplasmic reticulum, mitochondria. And then these are not real super resolution localizations, but what we envisage it will look like when we're able to incorporate the single molecule data into the EM models. So again, this is an elephant in the room. So both of these modalities are going to chuck out huge amounts of data, and we really don't know how to bring these two data sets together in a meaningful way that will generate new biological knowledge yet. But we've been awarded a Chan Zuckerberg Initiative grant to do that and to build it in a way that the resulting pipeline can be installed in any light microscopy lab without having to have an expert electron microscopist to run it. And for this project, we'll be working with Paul French at Imperial College with his Easy Storm system, and Ricardo Enriquez for his expertise in the data analysis, and also with Martin Jones from Microscopy Prototyping and Amy Strange from our scientific computing team. And we'll be installing the whole pipeline in Ben Luz's lab in Stellenbosch, who's also a member of Global Bioimaging, so that we can make sure that it really works before we roll out to other teams that don't have EM experts. So that's pretty much it. If you are interested in the volume EM techniques that I've talked about, then you can use this QR code, which will send you over to the new Volume EM community website. There's a bit of explanation about the techniques. There are plenty of contacts there, and if you want to get involved in the community, then you can join one of our working groups. And I'd like to, so Aaron is just about the only person in the lab who I didn't show work from today. So otherwise you've seen the whole of the EMSTP team. We're going to be advertising for positions on the Chan Zuckerberg grant shortly, so please look out for those adverts if you would like to join Paul, Ricardo and I in this amazing new challenge. We'd like to thank the funders and the groups at the CRIC that we work with, and I'll leave this just to play. So this is all of the volunteers who contributed to our first citizen science project and help us to train the machine learning algorithms to build the nuclei. And thank you very much for your attention. I can stop that. It goes on for quite a long time. There's 6,000 names on it.
Thank you for this fascinating talk. Other questions in the audience? So I have one question about the citizen science way of working, because you went from very long time required for analysis to drastically reduced pipeline. Does this scale, can you transfer this knowledge to other setups, or is it very so specific to your precise microscope that if someone in another place would want to do it, it would have to redo this whole process?
Yeah. So I think there's two different aspects there. So for the citizen science projects, anybody can build their own project on the Zooniverse. There's something called the project builder. So if you want to feed out your own microscope images and ask the general public to help you to analyze them, then you can do that yourself, but you can also contact us. So Helen has CZI funding to roll out citizen science throughout the community. She's actually on maternity leave at the moment, but if you're interested, drop me an email and I'll put you in touch with Martin, and we can provide some training material and give you some advice on building your own projects. And then the other aspect is, now that we have the algorithm, so the data is available, we've published the nuclear envelope paper, you can download the raw microscope data, you can download all of the segmentations. So microscope data is Empire, segmentations are on GitHub, and the algorithms are on GitHub. But what we haven't managed to do yet is make the algorithms available in a really user-friendly way, so our scientific computing team is working on that. And then there's a question of how transferable it is to different data sets. So we're now running the nuclear envelope algorithm over about 20 old EM data sets that we have at the CRIC that are collected on different types of volume electron microscope with different cells and different tissues. And we're finding that it generalizes very well. it makes the same mistakes for the most part. So sometimes it mistakes the nuclear envelope for the plasma membrane for the nuclear envelope. Sometimes we have a bit of missing nuclear envelope. So we're also building a platform where users can do a little bit of correction, which would then retrain the algorithm for application-specific use to try and make it really user-friendly to port to other data sets.
Okay, thank you. Do you have other questions? Okay. Well, that's it. Thank you very much for the talk. We're right on time, so it's perfect.
N'oubliez pas qu'il y a le concours des photos. Don't forget there is the photo contest, so send your beautiful, funny or whatever images to the email addresses that were communicated to you recently by email.
Yeah. I just wanted to advertise that we have a serial block face and the red tomography in workshops. It's in the Zeiss brought SEM. So if you want to see this technique, discuss with experts, it's in Zeiss, you have a SEM, and you can see this both technique live. We brought samples and you can see them. So thanks a lot.